The first sign is not a creature. It is a flaw in the darkness. A thin silver string rises from the seafloor, bends slightly, breaks into smaller beads, and keeps climbing. The camera is almost motionless. The remotely operated vehicle hangs above a slope of mud and scattered shells, its lights pushing a small amber room into an ocean that has never known morning. Beyond the reach of the lamps, there is no blue, no green, no horizon. Only a depth so complete that the mind gives up trying to measure it. Then another thread appears, and another. The bubbles are not violent. They do not roar out of the ground like steam from a volcano. They lift themselves from the sediment with an almost delicate patience, as if the planet is exhaling through pores too small to see. A little distance away, pale mats lie over the mud like frost. Mussels crowd together in dark heaps. A few pink anemones stand open in the current. Something thin and white curls in and out of a crack. This is not a reef, at least not in the way the word usually feels. No sunlight falls here, no seagrass sways, no warm shallows flash with fish. The water is cold enough to make time seem slower. The pressure is heavy enough to fold ordinary objects inward. And yet, the seafloor is not empty. It is busy, but in a hidden register. It is feeding on a leak. A pilot watches the screen aboard a ship far above, where waves slap the hull and coffee grows stale in paper cups. The scientists lean closer. On the monitors, a place that had been only a rough shape on a map becomes particular. Mud, shells, rock, a shimmer in the water, a small, persistent escape of gas from somewhere beneath the ocean floor. Methane. A gas we usually imagine as invisible, useful, dangerous, trapped in pipes, burning blue on a stove, gathering in mines, rising from wetlands, threaded through the language of energy and climate. But here, it comes from under the seabed, from dark layers of buried carbon, from pressure, heat, microbes, ancient sediments, and faults that let the deep earth whisper into the ocean. What makes the scene strange is not that methane exists down here. It is that life has learned to gather around it. The camera drifts lower. The bubbles wobble past the lights. A mussel shell opens by the smallest fraction. On the white mat, bacteria are making a world out of chemistry. Somewhere under the mud, other microbes are consuming the very gas that is trying to escape. The seep is both a wound and a filter, both a door and a guard. It lets methane rise, and then it surrounds the rising methane with organisms that eat, transform, bury, and breathe it back into the planet's slow circulation. The ocean floor is often described as a boundary, the bottom of things, a final surface where whatever falls from above comes to rest. But at a methane seep, the bottom is not a bottom. It is an opening. And there are thousands of these openings. Some hide along continental slopes where shelves drop toward abyssal plains. Some line the edges of submarine canyons. Some sit on fault zones where rock has cracked under the stress of moving plates. Some appear as quiet pockmarks in soft sediment. Some gather around salt structures in the Gulf of Mexico, where dense brine collects in pools that look like lakes beneath the sea. Some flare in Arctic fjords where the water is shallow enough for the question of atmosphere to become uneasy. Some, only recently seen, exist at depths once thought too extreme for abundant animal communities, far down in hadal trenches where the pressure is almost beyond imagination. The places where the ocean floor leaks methane are not all alike. Some are small enough to be missed by a single survey line. Some run for hundreds of meters, Some release bubbles for decades. Some may flicker, sleep, and awaken again. Some are surrounded by clams, worms, mussels, bacterial veils, carbonate ledges, and crabs moving carefully through chemical gradients that would be toxic to many forms of life. Others leave only a mark after the leak fades, a hard patch of carbonate rock, a graveyard of shells, a roughness on the mud where corals may later settle and begin another chapter, one no longer powered by methane, but built on what methane left behind. To follow these places is to follow a story that does not begin at the seafloor. It begins higher, with the patient fall of life from the surface ocean. Every day, across the open sea, microscopic plants bloom in the light. They take carbon from the water, build soft bodies, divide, drift, and die. Some are eaten. Some are recycled before they sink very far. But a fraction of that living matter escapes the upper ocean and starts falling. It becomes marine snow, a dim shower of flakes, fecal pellets, shells, dead cells, fragments of bodies, and the invisible chemistry of lives that lasted perhaps only hours. Most of it is consumed on the way down. The deep ocean is not a place where food simply arrives untouched. It is inspected, bitten, dissolved, digested, repackaged, and scavenged through kilometer after kilometer of water. But some reaches the floor. Some is buried quickly by more sediment. Some enters layers where oxygen disappears, where ordinary decomposition gives way to older, quieter metabolisms. Down there, in the darkness below the mud, microbes continue their work. They do not need drama. They do not need speed. They need carbon, time, and the right chemistry. In oxygen-poor sediments, communities of microorganisms break down organic matter in stages. The most energetic reactions are used first, then the next, then the next. Eventually, in deep buried layers where other options have been spent, some microbes produce methane. This is biogenic methane, made not by fire, but by life, in a world without light. Elsewhere, methane forms through heat. As organic-rich sediments are buried more deeply over millions of years, pressure and temperature reshape them. The soft remains of organisms become hydrocarbons. Oil, gas, complex mixtures that migrate upward if rock layers, fractures, salt bodies, or faults give them a path. This thermogenic methane carries the signature of depth and time. It may begin far below the seep itself, in basins whose histories are written in the stacking of ancient seafloors. In many places, these sources mingle. Microbial methane and thermogenic methane travel together. Fluids carry salts, sulfide, heavier hydrocarbons, and dissolved gases. The seafloor is not a sealed lid. It is more like a long, compacted skin over a body that is still changing. The leak begins when pressure finds weakness. A fault cuts through sediment. A canyon slices into a slope and exposes layers that had been buried. A salt dome pushes upward, bending and breaking the strata around it. A landslide opens a pathway. A hydrate layer becomes unstable. The fluid moves not because it has intention, but because buoyancy, pressure, permeability, and fracture networks allow it. It follows the architecture of the Earth. It gathers under seals. It slips through sand beds. It climbs along cracks. It pushes through mud grain by grain until, somewhere on the floor of the sea, a bubble appears. From the ship above, that bubble is almost nothing. A point of gas in a planet-sized ocean. But to a sonar system, thousands of bubbles can become a ghostly column. Modern multi-beam sonar does not only map the shape of the seafloor, it can also reveal the water above it. Sound pulses move outward, strike objects or density contrasts, and return. Bubbles scatter sound strongly. On a scientist's screen, a seep may appear as a pale flare, a vertical smudge, a strange plume rising from a slope that otherwise looks smooth. This is one reason so many methane seeps have been found only recently. They were not always invisible. We simply had not been listening in the right way, or with enough coverage, or across enough margins. The deep ocean has always contained these quiet vents, but our maps were incomplete, our lines too sparse, our instruments too blunt, our attention directed elsewhere. Then, one survey after another, the seafloor began to show its leaks. On the U.S. Atlantic margin, a region not once expected to be so full of seepage, mapping revealed hundreds and then more than 1,000 unique gas flares. They clustered mostly north of Cape Hatteras, along the slope between the coast and the deep ocean. The discovery changed not only a database, but a feeling. A passive margin, a place without the obvious violence of a subduction zone, was not passive in the way people had imagined. It had faults, eroded layers, canyons, buried hydrates, and fluid pathways. It had hidden motion.
There is something humbling about that. For years, the ocean off a busy coastline can be crossed by ships, fished, cabled, studied, defended, governed, named, and still hold beneath it an entire set of breathing places almost no one has seen. The surface can be familiar while the bottom remains unknown. A seep does not announce itself to the sky. A fishing vessel can pass above one and feel nothing. A city can glow on shore, its roads and towers arranged as if the world has been comprehensively measured, while below the outer shelf, a dark community lives around a chemical trickle from the sediment. That hiddenness is part of the subject. Methane seeps are not rare in the sense of being accidental curiosities, but they are difficult to know. They exist in the realm where geology, microbiology, chemistry, climate, and deep-sea ecology overlap. Each seep is a point where many systems touch. The carbon cycle becomes visible. The crust becomes porous. The deep biosphere reaches the sea floor. Animals without access to sunlight build bodies from bacterial partnerships. The ocean becomes an active participant, dissolving, oxidizing, transporting, and sometimes preventing methane from ever reaching the air. The phrase, "the ocean floor leaks methane," sounds simple. But the leak is not simple. A bubble rising from mud has already passed through a long sequence of gates. It may have begun as a molecule produced by a microbe in a buried pore space. It may have been trapped for a while in a cage of water molecules, frozen into methane hydrate, sometimes called fire ice because it looks like ice and can burn when brought into human hands. It may have moved through sediments so slowly that a human lifetime would be too short to watch its journey. It may have crossed zones where other microbes tried to consume it before it escaped. It may enter the water column only to dissolve and feed still more bacteria. It may never reach the surface. It may become carbon dioxide in the deep ocean. It may become part of a clam, a mussel, a mat, a carbonate crust. What looks like escape is also transformation. A cold seep is called cold, not because it is always literally colder than the surrounding water, but because it is not a hydrothermal vent. It is not driven by scalding volcanic fluid pouring out of newly formed crust. Its fluids may be near ambient seawater temperature, sometimes only subtly different from the water around them. The energy here is chemical, not thermal. It is the energy stored in reduced compounds, especially methane and hydrogen sulfide, meeting microbes capable of using them.
In the sunlit ocean, plants and algae take light and make organic matter. At seeps, bacteria and archaea use chemical reactions instead. This difference changes everything. It means an ecosystem can root itself in darkness, not as a scattered community waiting for food to fall from above, but as a local abundance built from below. It means the seafloor can host islands of density in an otherwise food-limited world. It means a place that would seem hostile by ordinary standards can become crowded. The first colonizers at many seep sites are microbial mats. They look, under the lights of a vehicle, like white or yellowish fabric spread over the sediment. Sometimes the mats ripple gently in the current. Sometimes they surround bubble streams. Sometimes they mark where sulfide-rich fluids are close to the surface. To the eye, the mats can appear simple, almost blank. But they are not blank. They are living chemistry made visible. Beneath and within them, methane is oxidized. Sulfate from seawater is reduced. Sulfide is produced. Carbonate can precipitate. The sediment becomes a layered chemical landscape. A few centimeters may separate one world from another. Oxygenated water above, anoxic mud below, sulfide at one depth, methane at another, microbes arranged around gradients as carefully as forests arrange themselves around light and water on land. Over time, these microbial processes can help create rock. Carbonate forms around methane-derived carbon. It hardens the seep site. What began as a leak in mud can become a ledge, a pavement, a chimney-like structure, a rough outcrop on which other organisms can attach. The seep builds its own architecture. It creates the foundation for later life, and when the active seepage weakens, those stones may remain.
There's a strange patience in this. Methane rises, microbes feed, carbonate gathers, mussels settle, worms grow, shells accumulate. The seep changes phase by phase. It is not a static habitat. It has succession, aging, death, and afterlife. Mussels at methane seeps often belong to groups that carry symbiotic bacteria in their tissues. These bacteria use methane, sulfide, or both, depending on the host and the conditions. The animal offers shelter, access, and transport. The microbes offer nutrition in a place where ordinary food is scarce. A mussel bed may look like a pile of shells, but it is also a city of partnerships. Every individual is partly an animal, partly a microbial farm, partly a negotiation with the chemistry around it. Tube worms do something equally remarkable. Some seep tube worms have no mouth, gut, or anus as adults. They live by housing bacteria within a specialized internal organ. Their red plumes reach into the water, taking in oxygen and sulfide. Their roots may extend into the sediment, gathering chemicals from below. They are animals arranged like bridges between seawater and mud, between oxygen and reduced compounds, between visible bodies and invisible metabolism. A field of them can look like pale grass growing on the moon, but each tube is a structure of dependence. The worm cannot simply leave and graze elsewhere. Its life is bound to chemical flow. If the seep shifts, if sulfide declines, if sediment is disturbed, if a trawl crosses the site, if drilling mud blankets the community, if hydrate mining someday arrives uncarefully, the ecosystem may not recover on human time. Many seep animals grow slowly. Some tube worms can live for centuries. Their stillness is not weakness. It is an adaptation to a world where energy arrives as a slow underground signal. The Gulf of Mexico is one of the places where this signal has been studied most intensely. There, beneath waters crossed by storms, oil platforms, shrimp boats, and ships,
the seafloor is shaped by salt. Ancient seas evaporated, leaving thick deposits of salt that were later buried under sediment. Salt behaves strangely under pressure. It can flow, rise, pierce, fold, and deform the layers above it. Around these structures, hydrocarbons migrate. Fluids climb, brine escapes. Methane seeps feed mussel beds and tubeworm fields. Dense saltwater pools gather in depressions so salty and heavy that they remain separate from the seawater above.
Under the lights of a submersible, a brine pool can look impossibly familiar. A shoreline, a rippling surface, a lake at the bottom of the sea. Fish may drift along its edge, mussels crowd the rim. The brine itself is often low in oxygen and rich in methane and sulfide. An animal that enters it may struggle or die, depending on the conditions. Yet the margin of the pool can be alive because chemical extremes create opportunities for organisms adapted to them. The boundary becomes productive precisely because it is dangerous. The Gulf seeps remind us that methane leakage is not only a bubble story. It can involve oil, brine, asphalt, carbonate, hydrate, faults, mud volcanoes, salt tectonics, and biological communities that differ from one seep to the next. Some sites are dominated by mussels, some by tube worms, some by clams, some by microbial mats. The chemistry chooses the first language, and life answers in dialects. In the Pacific Northwest, along the Cascadia margin, the story changes again. Here, an oceanic plate is moving beneath North America. Sediments are scraped, compressed, folded, and fractured in a subduction zone that stores tension for earthquakes. Methane is held in sediments and hydrates along the margin. Bubble plumes rise from places like Hydrate Ridge and other seep fields offshore Oregon, Washington, and Northern California. Sonar surveys have revealed thousands of bubble streams across this broad region, many of them unknown before modern mapping made the water column visible. To imagine Cascadia only as a future earthquake zone is to miss part of its present life. The same tectonic forces that deform the margin create pathways. Compression squeezes fluids. Faults focus flow. Methane hydrates form where pressure is high and temperature low enough for water molecules to cage gas. At the feathered edge of hydrate stability, small shifts in temperature or pressure can matter. Scientists study these areas not because every bubble is a catastrophe, but because they are sensitive boundaries where climate, geology, and ocean chemistry may speak to one another. A hydrate is a peculiar thing. At the molecular scale, water forms a crystalline cage around gas, usually methane. It is solid, like ice, but it is not simply frozen water. It belongs to a pressure-temperature world. Bring it out of that world, and it can fizz, crack, dissociate, release gas, and vanish. On the seafloor, exposed hydrate may appear white, chalky, or translucent, sometimes covered by sediment, sometimes inhabited by specialized organisms. In the Gulf, worms have been found living on hydrate mounds. In other places, hydrate lies buried, inferred through seismic reflections, cores, temperature, chemistry, and the behavior of seeps above. Globally, gas hydrate stores an enormous amount of methane, but it is not a single tank waiting to empty. It is patchy, layered, constrained by depth, sediment, temperature, pressure, and geological history. Some hydrate is deep and insulated. Some lies closer to the edge of stability. Some is beneath Arctic shelves affected by past sea level change and present warming. Some is on continental slopes where bottom water temperatures shift slowly. The danger is not a cinematic switch that turns on everywhere at once. The danger is more subtle.
local destabilization, changing seepage, ocean acidification in deep waters, unknown feedbacks, and the difficulty of measuring a process scattered across the planet's submerged margins. The ocean itself is an important filter. When methane leaves the seabed, much of it dissolves before it can rise far. Bacteria in the water column consume methane using oxygen. In sediments, anaerobic microbes consume methane with sulfate and other electron acceptors before the gas escapes. These biological filters are powerful. They are one reason deep methane seeps are not usually direct chimneys to the atmosphere. By the time the methane has crossed the sediment and water column, much of it has been transformed. But the filter is not magic. It has limits. Depth matters. Bubble size matters. Water temperature, currents, oxidation rates, the strength of seepage, and the distance to the surface all matter. In deep water, methane usually has a long journey, and that journey gives microbes and dissolution time to work. In shallow seas, fjords, shelves, and Arctic coastal areas, bubbles may reach the surface more easily. The same molecule can have different fates depending on where it emerges.
This is why the Arctic draws so much attention. Around Svalbard, gas flares have been observed in fjords and along continental margins. Some occur in relatively shallow water. Some are linked to faults, organic-rich source rocks, fractured formations, and possibly the changing stability of gas hydrates or subsea permafrost in certain settings. The Arctic is warming rapidly, and shallow systems do not have the same water column buffer as deep slopes. Scientists are careful because uncertainty here is large. Not every plume is new. Not every plume is caused by modern warming. Geological seepage can be ancient, persistent, and natural. Yet the question remains urgent because the region is changing quickly, and methane is a greenhouse gas with a strong warming effect over shorter timescales. A scientist looking at Arctic bubble flares must hold two truths at once. The first is that methane seeps are part of Earth's natural carbon cycle, older than industry, older than measurement, older than the names on our maps. The second is that a warming world can disturb natural stores and pathways, especially where ice, permafrost, shallow seabeds, and hydrate stability overlap. The difficulty is not choosing the dramatic truth over the cautious truth. The difficulty is staying with both. In the deep ocean, drama usually arrives slowly. A seep field can sit undiscovered for thousands of years, feeding generations of animals no human has seen. Then, a ship passes overhead with the right instruments, and the place enters the record. A column of bubbles becomes data. A white patch becomes a target. A target becomes a dive. A dive becomes a video feed in a dark control room. People who have spent years studying chemical gradients suddenly fall silent because the seafloor has become visible. There is a particular kind of awe that belongs to exploration by camera. No one touches the place at first. It appears through light, distance, and delay. The ROV hovers. Thrusters stir a faint cloud. Lasers draw two green points on a shell bed to measure scale. A manipulator arm unfolds slowly, more like a cautious animal than a machine. The pilot lowers a sampling tube toward a bubble stream. The scientists speak in clipped, practical phrases. More left. Hold position. We are seeing active flow. Note the muscles. Can we zoom on that, Matt? Watch the current. Behind every calm sentence is the fact that the ship is floating above kilometers of water, over a hidden ecosystem assembled around a gas that most people encounter only as fuel or fear. A methane seep is not pretty in the ordinary way. It may be muddy, pale, crowded, dim,
chemically harsh. The animals are often white, cream, gray, red, or dark brown. There is little of the tropical palette that teaches people to love the ocean from a postcard. But the beauty of a seep is not decorative. It is structural. It lies in relationships. It lies in the way a mussel carries bacteria that carry the chemistry of the sediment into animal flesh. It lies in carbonate rock formed from microbial transformations. It lies in a bubble that is both a sign of escape and a possible meal. It lies in the fact that life is not waiting passively for sunlight's leftovers, but building an economy around the planet's buried breath. The leak is a place. The leak is a history. The leak is also a question. How much methane is moving from sediments into the ocean? How much is consumed? How much becomes carbon dioxide in deep water? How much reaches the air in shallow settings? How does seepage change with storms, tides, earthquakes, landslides, warming currents, hydrate dissociation, or the slow migration of fluids through rock? How old are these communities? How connected are they across ocean basins? Can larvae travel from one seep to another through a dark sea where suitable habitat may be separated by vast distances? What happens when a seep dies? What happens when a new one opens? The questions are difficult because seeps are scattered, variable, and often hidden. A bubble plume may pulse. It may release more gas at one time of day than another, more after a disturbance, less after a pathway clogs, more when pressure changes slightly, less when hydrate reforms. A single measurement may not represent a year. A single year may not represent a decade. The seafloor can breathe irregularly.
To understand it, scientists leave instruments behind. Landers settle on the bottom with cameras, chemical sensors, hydrophones, bubble counters, and sometimes rotating sonars that watch a seep in place. They record when bubbles emerge, how often, how large, how fast. Some instruments listen because bubbles make sound when they form and break apart. A seep can have an acoustic signature. Its tiny bursts can be translated into frequency, strength, rhythm. The ocean floor, in its own way, can be heard leaking. This is one of the quiet revolutions in ocean science, the move from visiting to observing. A dive gives a moment. A lander gives a record. A mooring gives seasons. Repeated mapping gives change. Chemistry gives rates. Isotopes give origins. DNA gives hidden communities. Seismic profiles give pathways beneath the mud. Together, the seep becomes less like a spectacle and more like a system. Still, there is always more unknown than known. The ocean is vast in a way that defeats easy confidence. Even now, many margins remain poorly surveyed. Many seeps have probably never been mapped. Some are too weak for bubble detection, but still chemically active. Some emit dissolved methane without obvious bubbles. Some may lie under ice. Some may be masked by rough terrain. Some may be known to animals and microbes for millennia before they are known to people. This incompleteness matters. For a long time, the deep seafloor was imagined as mostly uniform, slow, and empty, a dark plain receiving the debris of the upper ocean. Hydrothermal vents changed that picture in the late 20th century. Cold seeps deepened the change. They showed that the seabed is not just a graveyard of falling matter, but also a source. Energy can come from below. Life can concentrate around chemical seepage.
The deep ocean can contain oases, but not the kind made by sunlight and water in a desert. These are oases of reduced compounds, pressure, microbial talent, and geological leakage. The word oasis can be misleading if it makes the surrounding deep sea seem lifeless.
It is not lifeless. The abyss has animals, microbes, tracks, burrows, slow-moving predators, scavengers, suspension feeders, soft-bodied creatures, and patient communities adapted to scarcity. But a seep changes the density of possibility. It intensifies the local field. It provides a dependable source of chemical energy, and around that source, life becomes layered, crowded, and specialized. Imagine crossing a dark plain where food falls unpredictably from above. Then, imagine finding a village built around a spring that flows from underground. That is closer to the feeling of a seep. But it is not only a village. It is also a refinery, a filter, a cemetery, a nursery, a laboratory, and a monument to buried carbon. On the U.S. Atlantic margin, the realization that seeps were widespread came through the patient assembly of evidence. Survey lines, water column backscatter, ROV dives, seismic profiles, databases corrected for duplicate detections, named seep fields, clusters, depth distributions, geological interpretation. Places like Norfolk, Baltimore Canyon, Washington Canyon, Bodie Island, Blake Ridge, and others entered a map that is still incomplete. At the Bodie Island seeps, bubble streams rise from relatively bare sediment near bacterial mats and mussel shells. The scene is both modest and extraordinary. It does not look like a grand volcanic opening. It looks like the floor has begun to fizz. And perhaps that is why it is so compelling. It forces a change in scale. The Earth does not only reveal itself through mountains, earthquakes, eruptions, and trenches. Sometimes it reveals itself through a small stream of gas in cold water, a white film of bacteria, and the open shells of animals living by a chemistry older than sight. The Atlantic margin also challenges assumptions about where seeps should be. A passive margin is not where one expects the Earth to look restless. There is no plate diving beneath another, no obvious chain of volcanoes, no dramatic trench. Yet passive does not mean still. Sediments compact, canyons erode, salt moves, hydrates respond to pressure and temperature. Old faults can remain pathways. Fluids migrate along subtle routes. The slope may look calm at the surface of a bathymetric map, but beneath it, the architecture is complicated. The phrase passive margin belongs to plate tectonics. The seep belongs to everything that still happens after tectonic drama has quieted. Farther west, Cascadia offers the opposite feeling, a margin where the drama is hidden but expected. The seafloor offshore the Pacific Northwest is shaped by convergence. The Juan de Fuca plate moves beneath the continent. Sediments pile and deform. Earthquake hazards occupy the public imagination. Yet the methane seeps along this margin bring another dimension to the story. They reveal the chemical consequences of tectonic compression. They show that a subduction zone is not only a place of future rupture, but a place of present fluid flow. Hydrate Ridge, offshore Oregon, has become one of the emblematic sites for studying methane hydrate and seep ecosystems. There, methane-rich fluids feed microbial mats, clams, and other organisms. Bubbles rise. Hydrate sits in the sediments. Scientists measure chemical gradients, collect cores, image plumes, and ask how methane moves between buried deposits and seawater. The name itself feels almost too direct. Hydrate Ridge. A ridge shaped into science by the substance it holds.
The Pacific Northwest also teaches the importance of edges. Not coastlines, but stability edges. Gas hydrate exists only where pressure and temperature permit it. On a continental slope, that zone has an upper limit and a lower limit, a three-dimensional shape that cuts through sediment. If warming bottom water shifts that boundary, hydrate near the limit may dissociate. If sea level changes, pressure changes. If sediment warms from below or fluids move through it, the balance shifts. Seeps sometimes occur near these limits, but not always. The relationship is complex. Some seepage comes from sources unrelated to contemporary hydrate dissociation. Some hydrates are remnants of past conditions. Some seeps bypass hydrate altogether through faults. The ocean floor does not obey one explanation. It is a place where local history matters.
In the Gulf of Mexico, salt history matters. In Cascadia, subduction matters. In the Atlantic, canyons, faults, erosion, and hydrate geometry matter. In Svalbard, fjord geology, Arctic warming, shallow water oxidation, fractured bedrock, and organic-rich source layers matter. In Hadal trenches, the accumulation of organic matter in V-shaped depressions and the bending of tectonic plates matter. Each seep is a sentence written by its region. Together, they form a language of leakage. The newest chapters in that language have come from the deepest parts of the ocean. For years, scientists expected Hadal trenches to be unusual, but largely dependent on food falling from above. Carcasses, sedimented organic matter, landslide deposits, the faint rain of carbon from the surface. Trenches are places where the seafloor plunges into narrow, immense depths. They are cold, dark, pressurized, and difficult to reach. Their name comes from Hades, and the name has always carried a hint of myth. Then, submersibles began to show something more. In the Kuril-Kamchatka Trench and the Western Aleutian Trench, recent dives found widespread chemosynthetic communities associated with cold seeps at depths reaching more than 9 kilometers. Dense fields of tube worms and bivalves appeared, where methane-rich fluids reached the seabed. At one site, the depth was about 9,533 meters, deeper than Mount Everest is tall. The animals were not isolated oddities. The communities extended across large stretches of trench terrain, suggesting that Hadal seep ecosystems may be more common than once imagined. It is difficult to hold that image in the mind. A trench bottom under nearly 10 kilometers of water. Pressure so immense it would crush ordinary machinery. No sunlight, near freezing darkness. And there, at the boundary between accretionary prism and trench floor, life gathered around seepage from the mud. The geologic model is elegant in its severity. Trenches can trap organic matter. Material falls from productive surface waters, slides from slopes, and accumulates in the V-shaped depths. Under anoxic conditions, microbes generate methane in buried sediments. Compression, subduction, faults, and fractures help move methane-rich fluids laterally and upward. At the seafloor, the fluids escape, and the chemistry supports animals that do not depend on photosynthesis directly. This does not mean the surface ocean is irrelevant. The buried carbon may still begin with sunlight, with plankton, with organic matter falling or sliding into the trench. But at the seep itself, the immediate engine is chemical. The energy is released in darkness, mediated by microbes, embodied by animals adapted to one of the most extreme habitats on Earth. The discovery changes the emotional map of the deep sea. It reminds us that the places we call remote are not necessarily barren. It reminds us that pressure does not forbid complexity. It reminds us that life may occupy the seams of geology wherever chemistry permits.
The Hadal seep also changes the scale of methane seep thinking. Seeps are not only on accessible continental slopes, not only near energy provinces, not only at margins where ships commonly survey. They may exist in the deepest trenches, along tectonic fractures, under pressures that make direct observation rare and expensive. The inventory of seep life is likely still young. Every time a new seep field is found, the map becomes less blank and more unsettling. Not because the methane itself is always an immediate threat, but because the planet becomes more intricate than before. There are more pathways, more habitats, more microbial processes, more interactions between rock, water, life, and climate. The Earth is not a collection of separate spheres. It is a continuous exchange. And methane seeps are places where that exchange becomes visible. Consider one methane molecule. It might begin as carbon dioxide fixed by a diatom in surface water. The diatom dies, part of it sinks. It is buried on a slope, then buried deeper by more sediment. Oxygen vanishes. Microbes alter the remaining organic matter. Methanogens produce methane. The molecule diffuses through pore water, perhaps joins a bubble, perhaps enters hydrate, perhaps waits for centuries inside a crystalline cage. Later, a shift in pressure or a fracture releases it. It rises through sediment toward the sulfate zone. There, anaerobic archaea and bacteria oxidize it, consuming sulfate and producing bicarbonate and sulfide. The bicarbonate helps form carbonate. The sulfide feeds bacteria that feed a tube worm. Or the methane escapes as a bubble, dissolves into seawater, and is eaten by aerobic bacteria in the water column. Or it reaches shallow water, crosses into the atmosphere, and becomes part of climate. One molecule can pass through life, stone, water, and air. Not metaphorically, literally. That is why methane seeps matter beyond their strangeness. They are active nodes in the carbon cycle. They store carbon, move carbon, transform carbon, and sometimes release carbon. They create habitats and chemical sinks. They complicate climate models. They offer analogs for possible life in other ocean worlds where sunlight may be absent, but chemical energy may exist. They challenge simple divisions between geology and biology. In school diagrams, the carbon cycle often looks clean. Arrows connect atmosphere, plants, soil, ocean, rocks, and fossil fuels. The arrows are useful, but they are too smooth. At the seafloor, the arrows become mud, pressure, bacteria, shell beds, hydrate lenses, bubble plumes, carbonate crusts, and dissolved methane curling through cold water. The cycle is not only a diagram, it is a landscape. And like every landscape, it has weather. Not weather of clouds and rain, but variability. Seepage may respond to tides through pressure changes. It may respond to storms indirectly through waves and currents on shallower shelves. It may respond to earthquakes by opening or closing pathways. It may respond to sediment slumps. It may wax and wane as hydrate forms or breaks down. A bubble stream observed on one dive may be stronger on another. A patch of bacterial mat may expand or shrink. A mussel bed may grow old as fluid flow migrates away. The seep is not an object. It is behavior. This makes it hard to count methane from the sea floor. You cannot simply find every seep, measure once, and add. The system moves. The ocean removes methane while it moves. Microbes change rates with conditions. Plumes bend with currents. Bubbles dissolve as pressure drops. Some methane enters sediments. Some becomes biomass. Some becomes carbonate. Some becomes carbon dioxide. Some remains dissolved and travels with water masses. The deeper the seep, the more time the ocean has to intercept it. A bubble released at 2,000 meters must survive pressure changes, dissolution, microbial attack, and turbulence before it reaches the surface. Most will not. The methane may enter the deep water instead, where it can be oxidized over time. This is why many scientists caution against imagining deep hydrates as a simple atmospheric trigger. The ocean is a shield, though not an infinite one. In shallow water, the story is more direct. A bubble from a continental shelf or fjord has less distance to travel. In Arctic seas, where shelves can be broad and shallow, and where subsea permafrost or hydrate systems may be changing, the question becomes more urgent. Yet, even there, careful measurement is necessary. Seepage observed today may have existed long before modern warming. Methane can come from microbial sources, thermogenic sources, thawing permafrost, decomposing hydrate, or mixtures. Some emissions vary seasonally, some are focused by faults, some are blocked or consumed before reaching the surface. The truth has to be built site by site. That is one of the moral demands of this subject. It resists both indifference and panic. Indifference would ignore the fact that methane is powerful, that hydrates and subsea carbon stores are climate relevant, that shallow systems may change, and that deep-sea habitats are vulnerable to human disturbance. Panic would flatten the complexity into one catastrophic image, as if all seafloor methane were waiting to erupt into the sky tomorrow. The real story is slower, stranger, and harder. It is a story of buffering and leakage, stability and sensitivity, ancient processes and modern change. A methane seep can be natural and still important. It can be old and still vulnerable. It can be beautiful and toxic. It can release greenhouse gas and support rich life. It can be a source of carbon to the ocean and a sink through microbial oxidation and carbonate formation. It can feel like a threat and a sanctuary at the same time.
The more closely one looks, the less useful simple categories become. The human part of the story is also full of contradictions. We search for methane seeps with ships powered by fuel. We study methane hydrate partly because it may be an energy resource, and partly because disturbing it could be risky. We send robots into habitats that may be harmed by the industries that mapping makes possible. We describe deep ecosystems as alien, while our own demand for energy and minerals increasingly reaches toward them. We worry about methane as a climate gas, while using methane as a fuel. We marvel at seep organisms whose existence depends on hydrocarbons, while knowing that hydrocarbons have altered the atmosphere above them. None of this makes the science less necessary. It makes it more necessary. A place cannot be protected, modeled, or understood if it remains unknown. Yet, knowledge is never neutral in the deep sea. A map can guide conservation. It can also guide extraction. A sample can reveal a microbial process. It can also disturb an ancient community. A discovery can deepen wonder. It can also invite attention from forces that do not wonder. The seep sits at the boundary between curiosity and consequence. There are moments in deep-sea footage when this becomes palpable. The vehicle's lights sweep over a mussel bed, and the shells extend beyond the frame. The pilot pulls back, and still the bed continues. A crab moves across them with slow mechanical precision. A fish hovers at the edge. A white mat lies just beyond. Bubbles rise from the sediment in the background. Nothing is staged. Nothing is symbolic. And yet the scene seems to contain several of the largest questions at once. How does life begin around energy? How does the Earth store and release carbon? How much of the planet is alive in ways we rarely see?
What do we owe to places we discovered only because technology finally caught up with their darkness? The seep does not answer. It continues leaking.
If there is a philosophy in methane seeps, it is not a grand one. It is practical and physical. Boundaries are porous. Darkness is not emptiness. Waste becomes food. Poison becomes energy in the right metabolism. A gas moving upward can build rock. A leak can become a habitat. The bottom of the ocean can be a beginning. But even those statements are too polished compared with the actual place. The actual place is mud, current, shells, chemical gradients, bacterial filaments, instrument noise, thruster wash, uncertainty, and a bubble rising. It is worth staying with that bubble. At first, it is round and clear. As it rises, pressure decreases. It may expand slightly. Methane diffuses out. Other gases may diffuse in. Hydrate may form briefly on its skin under some conditions, a delicate shell slowing dissolution. The bubble may shrink if methane leaves faster than pressure expansion grows it. It may wobble, split, merge, or vanish into dissolved form. If it survives long enough, it may join a plume. If not, it becomes part of the water column's invisible chemistry. A bubble is not a container moving unchanged from bottom to top. It is an event. Its fate depends on physics and biology. The water is not passive. Microbes are not passive. Even the bubble's surface is a site of exchange. By the time a methane molecule has traveled a few meters, it may no longer be in the bubble. By the time the plume has risen through hundreds of meters, the gas and its chemical aftermath may be spread through a volume of water far larger than the seep itself. This is why visual footage can mislead as much as it reveals. We see bubbles and imagine release. We do not see dissolution. We do not see microbial oxidation. We do not see the dissolved methane carried sideways by currents. We do not see the carbon dioxide produced later. We do not see the isotopic signatures that tell the molecule's history. The camera gives the mystery a shape, but the chemistry continues beyond the frame. Scientific instruments extend that frame. A CTD rosette lowers bottles into the plume and closes them at chosen depths. Water samples come back carrying dissolved gases. Sediment cores preserve chemical gradients in mud. Push cores, taken by ROV arms, capture mats and pore water. Mass spectrometers measure gases in situ. Seismic tools image layers and faults. Autonomous underwater vehicles fly close to the bottom, mapping terrain with high resolution. Sonars reveal plumes. Hydrophones listen. DNA sequencing reveals organisms too small to see and relationships too intricate for the naked eye. The modern seep is discovered by a choir of machines. But the machines are not the story. They are how humans learn to be present where our bodies cannot easily go. The deep ocean resists the human body. Pressure, cold, darkness, distance, expense, and risk keep us away. A diver cannot swim to most seeps. A person inside a submersible can visit only a tiny patch for a limited time. Most of our knowledge comes through cables, sensors, robotic arms, shipboard labs, and images watched by people who remain suspended above the site by kilometers of seawater. This distance creates a strange intimacy. We can see a clam open on the sea floor while being unable to breathe the water above it, unable to touch it without machinery, unable to remain without a vessel. Perhaps that is why seep footage feels dreamlike. It is real, but mediated through an impossible act of looking.
One of the most haunting features of seep communities is their dependence on invisible flow. A coral reef can be understood by light, a kelp forest by sunlight, nutrients, waves, and rock. A seep community asks us to imagine energy moving through sediment, not from the sky, but from beneath the floor. The animals are arranged around something the camera cannot directly show: methane concentration, sulfide availability, oxygen penetration, sulfate reduction, fluid migration. The visible community is a map of invisible chemistry.
White mats mean one thing, mussels another, tube worms another, carbonate another, dead shells another, bare sediment with bubbles another. A trained observer reads the bottom as a field of clues, where methane is high, where sulfide reaches the surface, where flow has shifted, where a community is young, mature, declining, or replaced. A new seep may begin with microbes. Then come animals that can exploit the chemical setting. As carbonate forms, the habitat changes. When seepage weakens, the organisms tied most directly to methane and sulfide may die back. Their shells remain. Their tubes remain. The hard carbonate they helped create remains. Corals and sponges may colonize the exposed rock, feeding not on seep chemistry, but on particles carried by currents. The seep's afterlife supports a different community. The original leak becomes a foundation for organisms that do not depend on the leak itself. In this way, methane can shape life even after it stops flowing. The seafloor remembers. It remembers in rock, in shell beds, in isotopes, in pockmarks, in carbonate slabs, in the distribution of species, in old tubes standing like abandoned architecture. Geologists can read ancient seeps in exposed rocks on land, places where methane once rose through sediments long before those sediments were uplifted into cliffs and hills. Fossil seep communities show that this kind of habitat has existed through deep time. The modern seep is not an anomaly of the present ocean. It is part of a recurring planetary pattern. There were methane seeps when no humans existed. There will likely be methane seeps after human maps have changed beyond recognition. That deep continuity can feel calming, but it should not make the present feel harmless. Ancient processes can be altered by modern forcing. The fact that a system is natural does not mean it is immune to acceleration, disturbance, or damage. A forest fire is natural. A flood is natural. Methane seepage is natural. But context, rate, and vulnerability matter. Deep-sea mining, trawling, drilling, cable laying, and hydrate extraction all carry potential impacts. Cold seep ecosystems may be patchy and slow to recover. Many species are specialized. Some may exist only in limited regions. Because seeps are small compared with the ocean, they can be overlooked in broad management decisions. A trawl scar through a seep community might cross in minutes what took centuries to grow. A drilling accident could smother habitats that live by delicate chemical exchange. Mining in hadal or deep-sea environments could disturb communities before they are even described. There is an old assumption that depth protects. It does not protect as well as we once thought. The deep sea is increasingly reachable. Technology that allows exploration also allows exploitation. The same maps that reveal biological marvels can reveal mineral deposits, energy resources, and industrial opportunities. This does not mean exploration should stop. It means exploration must be accompanied by humility. A seep community should not become collateral damage simply because it lives far from shore and outside ordinary imagination.
The places where the ocean floor leaks methane ask for a slower ethic. Not sentimentality, attention. They ask us to know what is there before we decide what can be taken. They ask us to measure not only resources, but relationships. They ask us to remember that a dark habitat is not empty just because it is inconvenient to visit. There is another reason these places matter, one that extends beyond Earth. Astrobiologists are interested in chemosynthetic ecosystems because they show that life can flourish without sunlight when chemical energy is available. Hydrothermal vents are often mentioned in this context, but cold seeps add another model. They are not the same as vents. They show that microbial metabolisms tied to methane, sulfide, and other reduced compounds can support complex animal communities in cold, dark environments. On icy ocean worlds where sunlight may never reach subsurface seas, chemical gradients could be among the few possible sources of energy for life.
It would be careless to say that methane seeps on Earth prove life elsewhere. They do not. But they expand the imagination of habitability. They remind us that sunlight is not the only way a biosphere can organize itself. They show that geology can feed biology. They show that an ocean can hide metabolic worlds beneath darkness. When scientists think about Europa, Enceladus, or ancient Mars, they often think about water, rock, chemistry, and energy. A cold seep is one earthly expression of those ingredients meeting. Still, the seep is not valuable only because it helps us imagine other worlds. It is valuable because it is here, because it is part of this planet's life, because in a dark ocean, under pressure, in mud and chemical gradients, organisms are doing the work of existence with a patience and strangeness that does not need cosmic justification. The more one learns about methane seeps, the harder it becomes to think of the sea floor as a surface. It is a membrane between worlds. Above it lies the ocean, layered by temperature, salinity, pressure, currents, oxygen, plankton, fish, storms, and human roots. Below it lies sediment, pore water, microbes, buried organic matter, hydrate, faults, salt, heat, pressure, and rock. At a seep, these worlds communicate. The communication is not loud, it is not always visible, but it is continuous enough to build communities, continuous enough to make stone, continuous enough to enter climate calculations, continuous enough that scientists cross oceans to find it. There is a scene that repeats in many seep expeditions. A ship arrives at a coordinate chosen from acoustic data. The crew deploys an ROV. The vehicle descends through the upper ocean, where daylight fades from blue to black. Particles streak past the camera. The water grows darker. The pressure increases. The sea floor finally appears as a pale shape below, first vague, then detailed. The vehicle levels out. The pilots begin a survey line. At first, there is only sediment. Ripple marks. A brittle star. A fish moving away from the lights. Then a white patch appears. Then shells. Then bubbles. The room on the ship changes. People lean toward the screens. Someone says, "There it is," not loudly, because the moment feels too delicate for loudness. "There it is." The phrase carries relief, excitement, confirmation, and the old human pleasure of a mystery becoming visible. But the seep was there before the ship arrived. The bubbles did not begin for the camera. The mussels did not gather to be discovered. The bacteria did not wait for a name. Science often frames discovery as a human event, and in one sense it is. A thing becomes known to us. It enters records, papers,
datasets, maps, and public imagination. But for the seep itself, discovery is an interruption. The community has its own continuity. The leak has its own timing. The animals have histories that do not require our arrival. This is important for the atmosphere of the subject. We are not entering empty darkness and creating meaning by looking. We are arriving late to places already meaningful in the language of metabolism, geology, and time. A methane seep does not perform wonder. It performs chemistry. The wonder is ours. The chemistry is older. When methane rises into sulfate-rich sediment, anaerobic oxidation becomes one of the key processes limiting methane emissions. The main actors are not visible animals, but consortia of archaea and bacteria, often working together in ways that still challenge simple descriptions of metabolism. They use sulfate to oxidize methane without oxygen, producing bicarbonate and sulfide. This process consumes methane before it enters the ocean, and it creates sulfide that can fuel other chemosynthetic bacteria. In that sense, the methane filter also feeds the community. A barrier becomes a source. A sink becomes a foundation. The same process that prevents methane from escaping freely helps produce the chemical energy used by seep organisms. This is one of the elegant circularities of seep ecosystems. The system protects the water column from some methane, while building habitat from methane's transformation. Carbonate precipitation is part of this. Bicarbonate produced during methane oxidation can combine with calcium to form carbonate minerals. These rocks often have isotopic signatures showing methane-derived carbon. They are geochemical records, but also physical structures. Microbes help lay the pavement on which later organisms live. The seep, then, is not merely a leak in rock. It is rock being made by a leak. It is carbon escaping and being fixed again in another form. It is a gas trying to rise and a community holding part of it down. This is why the question, "Is methane escaping from the sea floor?" is incomplete. The better question is, what happens to methane as it moves? Where is it produced? Where is it stored? Where is it consumed? Where is it transformed? Where does it enter organisms, minerals, dissolved pools, or the atmosphere? The path matters more than the presence. Methane in a buried hydrate layer is not the same as methane dissolved in pore water. Methane consumed by sediment microbes is not the same as methane crossing the sea-air interface. Methane feeding a mussel symbiont is not the same as methane oxidized to carbon dioxide in the water column. The molecule is the same. The consequence is not. For climate, this distinction is essential. Methane is a potent greenhouse gas in the atmosphere, but methane that is oxidized in the deep ocean before reaching the air has a different impact. It may contribute to deep water carbon dioxide and local acidification, but it does not warm the atmosphere as methane. This does not make it irrelevant, it shifts the concern. The deep ocean biofilter reduces one risk while creating or contributing to others that are harder to see. Ocean acidification is usually discussed as carbon dioxide entering from the atmosphere. But at seeps, carbon dioxide can also be produced locally by methane oxidation. In certain settings, this may affect deep-sea organisms, especially those living near the seep or on carbonate structures. The chemistry of the seep is not only a food source, it is also stress. Animals living there tolerate gradients that would exclude many others. Their success is not comfort, it is specialization.
Specialization brings vulnerability. A mussel adapted to methane-rich seepage cannot simply become a fish of the open slope. A tube worm dependent on sulfide cannot follow any current at random and survive. Larvae may disperse, but settlement requires the right chemical cues and habitat. The deep sea is enormous, yet suitable seep habitat is patchy. Connectivity among seeps becomes one of the central questions. How do specialized species find new sites? How far can larvae travel? How long can they wait? What happens when distances between active seeps are too great? To humans, the deep ocean looks continuous. To seep animals, it may be an archipelago, islands of chemistry separated by dark plains. Some islands flare brightly and fade. Some last centuries. Some are born after geological disturbance. Some vanish under sediment. Some are never found. This hidden archipelago may stretch across continental margins, trenches, basins, and polar shelves. The islands are not always mapped. Their inhabitants may be related across vast distances or isolated by geography and currents. The evolutionary story of seep organisms is still being written, often one dive and one genetic sample at a time. There is a quiet beauty in the way seep animals carry their worlds inside them. A mussel's gill tissue becomes home to bacteria. A tube worm's trophosome becomes a chamber of symbiosis. The animal is not an individual in the simple sense. It is a consortium, a habitat for microbes, a body built around cooperation. In the deep sea, where food scarcity can be severe, partnership becomes architecture. Evolution does not solve the darkness by giving the animal eyes alone. It gives the animal bacteria. The seep community makes visible a truth that is easier to forget in sunlit places. No life is solitary. Even at the human scale, we are microbial ecosystems. Forests depend on fungi. Corals depend on symbionts. Ruminants depend on gut microbes. At cold seeps, dependence is intensified and made dramatic. The animal's survival is tied directly to microbes that can turn chemical energy into living matter. The boundary between creature and environment becomes intimate. A seep mussel is not only living near methane, it is living with organisms that make methane useful. This is why the discovery of a seep community can feel like discovering a different answer to the question of what food is. Food is not only something that falls. Food can be made in darkness from a gas. Food can be a reaction. Food can be a partnership between a buried carbon molecule and a bacterium inside an animal under the sea. The phrase sounds almost mythic, but the process is biochemical. That is what gives it power. The wonder is not that science makes the world less mysterious. The wonder is that accurate description becomes stranger than fantasy. In the Gulf, mussels at brine pools live where methane-saturated fluids meet seawater. In the Atlantic, mussel beds cover carbonate and sedimented mounds. In Alaska and other northern sites, tube worm fields can stretch across the seafloor like pale vegetation. In hadal trenches, siboglinid worms and bivalves gather beneath crushing pressure. Each place repeats a theme without becoming identical. Methane rises, microbes respond, animals gather, rock forms. The theme is ancient, but the variations are local. A seep on a passive Atlantic margin does not feel like a seep along an active trench. A brine pool in the Gulf does not feel like a shallow Arctic flare. A hydrate ridge does not feel like a mud volcano. The word seep is a category.
but the actual places are more personal than categories allow. Some seep sites are named with scientific plainness. Others carry names that sound accidental, poetic, or strange. Hydrate Ridge, Blake Ridge, Pythia's Oasis, Norfolk Seeps, Bodie Island Seeps, Winter Sweet Valley. Names help humans hold a place in memory. They also reveal our need to domesticate the unknown just enough to return to it. Pythia's Oasis, off the Pacific Northwest, evokes an oracle. The name fits more than one might expect. Fluids emerging there from the seafloor have been interpreted as a window into conditions along the Cascadia megathrust, as if the margin is offering a small message from a locked fault system below. A seep can be ecological, chemical, and tectonic all at once. Its fluid chemistry may tell scientists about temperature, depth, and pathways. Its location may reveal fractures. Its flow may hint at pressures otherwise inaccessible.
The leak becomes information. Not all methane seeps are tied to hydrates. Not all are tied to faults. Not all are active in the same way, but each offers clues about subsurface processes. This is one reason geologists care about them. A bubble plume is not just a biological habitat. It is evidence of fluid migration. It says something about sediment permeability, pressure, gas generation, and structure. A seep is the visible endpoint of an underground route. To reconstruct that route, scientists combine evidence. Seismic profiles show layers bending, faults cutting, gas pockets disturbing reflections. Bathymetry shows pockmarks, mounds, scarps, canyon walls, and ridges. Chemistry distinguishes microbial methane from thermogenic methane by isotopes and associated hydrocarbons. Temperature and pressure define hydrate stability. Biology reveals sustained flow through the presence of seep-endemic communities. Carbonate ages can show how long seepage has occurred. No single clue is enough. The seep must be approached like a mystery, where every witness speaks a different language. The microbes speak through genes and isotopes. The animals speak through distribution. The rocks speak through minerals. The bubbles speak through sonar. The water speaks through dissolved gases. The sediments speak through pore water gradients. The challenge is to listen to all of them without forcing one explanation too early. This is a different kind of storytelling from the dramatic disaster narrative often attached to methane. It is slower, more complicated, and ultimately more compelling. The ocean floor is not a villain preparing a single event. It is a living geological boundary with many local behaviors. Some are stable, some are changing, some are poorly known, some are potentially sensitive. All deserve attention. The idea of methane hydrate as fire ice has captured public imagination because it seems impossible, ice that burns. A hand holding a chunk of hydrate can ignite the escaping gas as the solid breaks down. The image is memorable, but it can distort the scale and setting. In nature, hydrate is not a pile of campfire ice waiting on the sea floor everywhere. It is usually buried in sediment, shaped by local conditions, sometimes exposed at seeps, often inferred rather than seen. Its stability depends on pressure and temperature, and the response to warming can be slow because heat must penetrate sediment. Yet hydrate matters profoundly. It represents a large store of methane in marine sediments and permafrost regions. It influences slope stability, fluid flow, carbon cycling, and potential energy resources. Its dissociation can feed seepage. Its formation can trap gas and alter sediment strength. Hydrate-bearing sediments may be linked to submarine landslides in some contexts, though the relationships are complex. Hydrate is not merely a curiosity. It is part of the mechanical and chemical fabric of continental margins. There is an unsettling elegance in the hydrate cage. Water, the substance of oceans, traps methane, a gas born from carbon, under pressure in the cold. The cage holds until conditions change. Then it releases. In this form, the ocean stores a gas beneath itself, not in a metal tank or cavern, but in the microscopic geometry of ice-like crystals. The planet has many ways of hiding things. At seeps, the hidden becomes visible. Sometimes hydrate lies exposed as white chunks or crusts. Sometimes bubbles emerge from beneath hydrate layers. Sometimes animals live around it, and in rare cases, on it. The boundary between solid hydrate and seawater becomes active, gas escaping, microbes colonizing, worms feeding, sediments shifting. A piece of hydrate is not inert in such a place. It is temporary architecture. Temporary can still mean years, or decades, or longer. The deep sea changes at scales that do not match human impatience. A seep may seem stable during a research program, but be transient geologically. A tube worm may outlive generations of scientists. A hydrate lens may form and dissociate, with changes slow enough to be missed without long records. A bubble plume may vary hourly. Multiple clocks tick at once. This is part of what makes methane seeps cinematic. Not because they are fast, but because they layer time. A live video feed shows seconds. A mussel bed shows decades. A carbonate outcrop shows centuries to millennia. Sediment layers show millions of years. The methane molecule may carry ancient carbon, while the bubble rises in a moment. The camera watches the present. The seep contains the past. A slow documentary about methane seeps cannot really be about methane alone. It has to be about time. About how the ocean stores time in mud. About how life uses what the earth releases. About how the present climate meets ancient carbon. About how human observation briefly illuminates processes that continue in darkness. The cold open of a bubble stream is compelling because it is small enough to see and large enough to imply everything else. One thread of gas rising in the dark. Behind it, buried life, dead plankton, microbial metabolism, sediment compaction, faults, pressure, hydrates, oxidation, symbiosis, climate, exploration, and the unknown extent of the seafloor's leaks. A bubble is a door scaled down to a point. Through it, the story widens. The first widening is geological. Continental margins are the primary stage for many methane seeps because they collect sediment. Rivers deliver material from land. Ocean productivity adds organic matter from above. Slopes bury it. Tectonics deform it. Over time, thick sedimentary piles become both source and seal. Methane generation, migration, and storage become possible. Where pathways reach the surface, seeps appear. Margins are not all the same. A passive margin, like much of the U.S. Atlantic, forms where a continent has rifted and drifted away from another, leaving a broad shelf and slope without active subduction. Sediments accumulate over long periods. Canyons carve into the slope. Salt may deform layers in some regions. Old faults may persist. Hydrates may form at suitable depths. An active margin, like Cascadia or parts of Japan, Chile, Alaska, and New Zealand, is shaped by plate convergence. Sediments are compressed, accreted, faulted, and sometimes dragged downward. Fluids are squeezed out. Earthquakes and deformation create pathways.
Methane can move through the accretionary prison. Transform margins, back arc basins, rifted basins, and salt provinces each bring their own architecture. The ocean floor leaks where geology allows it.
This is why the map of methane seeps is not random. It follows the history of basins, the supply of organic matter, the depth of the water, the thickness of sediments, the temperature below the floor, the presence of gas hydrates, and the cracks through which fluids move. To look at seep distribution is to see the hidden skeleton of continental edges. But even a good map is incomplete. The seafloor is enormous, and survey coverage is uneven. Wealthy nations have mapped some margins intensely, while others remain sparse. Areas near energy development may be better studied for reasons that are not purely ecological. Deep trenches are hard to reach. Polar regions are difficult and seasonal. Some seeps produce no bubbles detectable in the water column. Some seepage is diffuse. Some habitats may be buried under sediment or hidden beneath ice. The unknown is not a gap around the edges. It is woven through the subject. The second widening is biological. Methane seeps show that life is not merely adaptable in a vague, inspirational sense. It is metabolically inventive. Microbes have evolved ways to use chemical gradients that animals cannot use directly. Animals have evolved ways to host those microbes. Communities have evolved around patchy, toxic, energy-rich environments. Evolution has not erased the harshness of seeps. It has learned to work within it. At a seep, methane and hydrogen sulfide can be abundant, but both are problematic for many organisms. Sulfide interferes with respiration. Low oxygen limits many animals. The chemistry can shift over centimeters. Sediments may be unstable. Yet, seep organisms not only survive, they build dense populations. Their success depends on managing toxicity and access at the same time. They must live close enough to chemical energy to benefit, but not so close or unprotected that it kills them. This balance gives seep communities their peculiar arrangement. Bacterial mats lie where sulfide reaches the surface. Mussels position themselves where methane and oxygen can both be accessed. Tube worms bridge sediment and water. Clams may bury into sediments. Mobile animals graze, scavenge, or prey around the edges. Some species are endemic to seeps. Others are visitors drawn by food or structure. Predators come. Opportunists come. The seep radiates effects into the surrounding benthos. It is not isolated from the broader ocean. Currents carry larvae away. Particles fall from above. Fish visit. Carbon moves outward. When seep animals die, their shells and tissues feed other organisms. The seep is a local concentration, but it remains connected. The third widening is chemical. Methane oxidation, sulfate reduction, sulfide oxidation, carbonate precipitation, hydrate formation, and gas dissolution all occur in overlapping zones. The chemistry is not merely background. It is the engine, the architecture, the hazard, and the record. A seep is a chemical landscape where organisms occupy niches defined by molecules. This is difficult to film. A camera sees shape and motion. Chemistry has to be inferred, sampled, measured. But sometimes chemistry becomes visible through biological color. White bacterial mats reveal sulfide oxidation. Orange or yellow mats may suggest different microbial communities. Black sediment can indicate anoxia and sulfide minerals. Carbonate crusts reveal long-term methane oxidation. Bubble plumes reveal gas escape. Brine pools reveal salinity so high that a separate underwater surface forms. At a seep,
the invisible keeps becoming visible indirectly. The fourth widening is climatic. Methane is a greenhouse gas, and seafloor methane stores are large. That fact can make every bubble feel ominous. But the climate role of marine methane seeps depends on pathway and fate. Deep seeps are often strongly buffered by dissolution and microbial oxidation. Shallow seeps may contribute more directly to the atmosphere. Hydrate dissociation under warming may occur in some upper slope and Arctic settings, but global catastrophic release scenarios are not the mainstream scientific picture for the near term. The more serious work lies in measuring actual fluxes, understanding vulnerable regions, and incorporating microbial filters into models.
The climate story is not an explosion. It is a set of thresholds, buffers, leaks, and uncertainties. This may be harder to communicate, but it is more truthful. The deep ocean is not a movie trapdoor. It is a chemically active reservoir interacting with climate in ways that can be slow, local, and still consequential. The fifth widening is human. Methane seeps sit close to questions of energy, extraction, conservation, and knowledge. Gas hydrates have been investigated as possible energy resources by several countries. Offshore drilling already intersects with seep-rich regions. Deep-sea mining may expand into habitats we barely understand. Climate change may alter seep systems, while human industries seek the same margins for resources. The scientific desire to know cannot be separated from the political need to decide. A voiceover can linger on a bubble, but policy will ask for boundaries, impact assessments, protected areas, baselines, monitoring, and responsibility. The seep becomes part of governance. And yet, despite all this, there is still the simple image: bubbles rising in the dark. A good story must keep returning there, not as a repeated trick, but as an anchor. The bubble is where abstraction becomes physical. Without it, methane is a number, a molecule, a climate term, a fuel. With it, methane becomes an event in a place. A place has texture. The mud may be soft enough for a sampling core to slide in with little resistance, or it may be crusted by carbonate. The bubbles may come from a crack, a small hole, or a broad shimmering patch. The water may carry flakes of bacterial material. Shells may be intact, broken, living, dead, or cemented. A fish may rest nearby, indifferent to the scientific importance of the scene. The ROV's lights may catch a shrimp's eyeshine. The manipulator arm may stir sediment and briefly erase visibility. The pilot may back away and wait for the water to clear. Such details matter because they prevent the subject from becoming only explanatory. The deep sea is not a concept. It is a place where machines get stuck, samples fail, currents push, cables strain, batteries matter, and a bubble can drift out of frame before anyone is ready. The practical reality of seep research is full of patience and frustration. Weather can cancel dives, instruments can misbehave, acoustic targets can turn out not to be active seeps. A suspected plume may be fish or noise. A core can miss the chemical hotspot by centimeters. A bubble sampler can clog. A lander can sink too deeply into mud. A ship can spend days transiting to a site only to find conditions unsafe. The ocean does not care about schedules. This is another reason the data are precious. Every confirmed seep, every sample, every time series, every video transect carries the cost of reaching a hidden place. Behind a clean map of dots are nights of survey lines, equipment checks, rough seas, software, fatigue, and decisions made in dim control rooms.
Maybe that is why scientists often sound careful when speaking about methane seeps. They know how much uncertainty sits behind each confident-looking statement. They know the ocean can overturn assumptions. The Atlantic margin did. The Hadal trenches did. The Svalbard fjords continue to complicate the climate question. Cascadia reveals new plumes as mapping expands. The map of known seeps is not the map of actual seeps. It is the map of our looking. And our looking has only recently become extensive enough to surprise us properly. To understand the places where the ocean floor leaks methane, it helps to imagine them as thresholds rather than vents. A vent suggests a pipe. A threshold suggests exchange. At a seep, buried carbon crosses into ocean water. Microbial life crosses from invisible process into visible mat. Geology crosses into ecology. A gas crosses into food. A fluid pathway crosses into a habitat. The deep past crosses into present chemistry. Human knowledge crosses from speculation to image. Thresholds are often quiet.
They do not always look important. A seep can be nothing more dramatic than cloudy sediment, a patch of white bacteria, and a few bubbles. But at such places, the Earth is negotiating with itself. What remains buried? What is released? What is consumed? What becomes stone? What becomes life? What reaches the air? These questions are not rhetorical for the planet. They are physical processes happening continuously, unevenly, and at scales from micrometers to ocean basins. A methane seep's smallest scale is microbial. In a sediment pore, a space between grains of mud, chemistry changes over fractions of a millimeter. Methane concentration rises. Sulfate diffuses downward from seawater. Archaea and bacteria form consortia. Enzymes catalyze reactions. Electrons move. Energy is conserved. Cells divide. Minerals precipitate. To us, the pore is invisible. To the microbes, it is the world. The largest scale is planetary. Methane generated and stored in marine sediments is part of the global carbon cycle. Hydrate stability is influenced by ocean temperature and sea level. Methane oxidation affects ocean chemistry. Atmospheric methane affects radiative forcing. Continental margin processes influence climate over long timescales. The bubble and the planet are connected through chemistry. This is the scale shift that gives the topic its emotional depth. From pore to planet. From microbe to margin. From a shell bed to the atmosphere. From a buried molecule to a warming world. A seep contains all these scales without announcing them. It simply leaks. When the ROV leaves, the lights rise away. The bubble stream continues in darkness. The muscles close and open. The mats continue their chemical work. The current carries dissolved methane sideways. The ship moves on to another station or returns to port. Data begin their second life in laboratories and papers. The sea floor resumes being unseen. This unseen continuation is important. We often confuse observation with existence. The camera turns off and the image ends. But the seep does not. It is still there after the live stream, after the publication, after the map is archived. It may continue through storms above, through nights and seasons, through migrations of fish, through the slow decay of shells, through sediment settling over old tubes. The deep ocean is full of ongoing things. Our glimpses are brief.
The places where methane leaks from the ocean floor are among those ongoing things. And perhaps their greatest lesson is that the planet is active in registers beyond ordinary attention. Not hidden because it is mystical, hidden because it is deep, slow, dark, and difficult. To say the ocean floor leaks methane is to say the Earth is not finished separating its layers. The buried does not stay buried absolutely. The living does not remain only at the surface. The air is not disconnected from mud. The crust is not disconnected from microbes. The deep is not disconnected from climate. Everything touches, but often through long, indirect paths. This can be unsettling. It can also be grounding. The planet is not a machine with clean compartments. It is a body of exchanges. Some exchanges are fast and visible: storms, waves, eruptions. Others are almost silent: diffusion, seepage, microbial oxidation, carbonate precipitation. Methane seeps belong to this quieter category. They are not less important because they are quiet. Their quietness may be why they were missed. Imagine the continental slope at night, though night is meaningless there because day never arrives. The water presses down. A bubble emerges. It rises a few centimeters, then another, then another. Around it, bacteria metabolize. A mussel bed filters water. A tube worm plume bends. A carbonate ledge holds. A fish passes through and disappears. No one sees it. No one needs to. The event is complete without witness. And yet, when witness arrives, something changes in us. We learn that the ocean floor is porous. We learn that life builds around leaks. We learn that a gas associated with danger, fuel, and climate is also the basis of hidden ecosystems. We learn that the deep sea is not a blank darkness beneath human affairs, but part of the same Earth system that includes our breath, weather, energy, and future. There are places on this planet where the boundary between geology and biology is so thin that it becomes meaningless to separate them. Cold seeps are among those places. The rock feeds the microbes. The microbes feed the animals. The animals shape the habitat. The habitat records the chemistry. The chemistry reveals the rock. A circle, but not a closed one. Carbon enters. Carbon leaves. Water moves. Life grows. Flow shifts. The seep ages. If the seep is strong and persistent, mussels and worms may thrive. If it weakens, the community may decline. If flow migrates a few meters, the living center of the habitat may move with it, leaving behind dead shells and new mats elsewhere. From a human perspective, this migration is subtle. From the animal's perspective, it is destiny. The seafloor is full of such destinies. A larva drifting through dark water may carry the potential to settle at a seep it has never known. It must encounter the right chemical cue, the right substrate, the right microbial environment. Many will not. Some will. A new generation begins where methane reaches the surface. The continuity of the species depends on hidden connectivity among scattered leaks. This is one of the most fragile aspects of seep life. Destroy one site, and the loss may not be easily replaced if nearby habitats are rare or disconnected. Protect one site, and it may serve as a source for others. But to design protection, we need to know where sites are, how communities connect, how long they persist, and what disturbances they can survive. Again, the science becomes practical. Wonder leads to responsibility. Not always directly, not always comfortably, but inevitably. The question of methane seeps is not only, what are they? It is also, how should we behave now that we know they are there? A cinematic journey can end in vastness, but the actual world continues into decisions. Shipping routes, fishing practices, drilling leases, protected areas, climate policy, research funding, seabed mining regulations, and energy choices all touch, however indirectly, the future of deep-sea seep habitats and the methane cycle. The seafloor may be far away, but it is not outside the reach of human consequence. Still, the ending should not turn into a speech. The subject deserves quiet. So, return to the bottom.
Return to a place where bubbles rise from mud under a black ocean. The ROV is gone now. The ship is a memory overhead. The data have traveled into human systems, but the seep remains in its own time. Methane moves upward through sediment. Some is consumed before it can leave. Some enters bubbles. Some dissolves. Some feeds microbes. Some helps form carbonate. Some supports animals whose bodies carry bacteria like hidden lanterns. The ocean presses from above. The earth presses from below. Between them, a narrow opening persists. Not a wound exactly, not a spring exactly. A seep. A small place where the planet fails to keep one of its secrets completely buried. In the dark, that failure becomes a world. The first time a person sees a methane seep on a live video feed, it can take a moment to understand the scale. The camera has no sky in it. There are no trees, no buildings, no human bodies, nothing familiar enough to make distance easy. The frame contains sediment and shells, perhaps a measuring laser, perhaps the arm of the vehicle. A mussel might be 10 centimeters long or 20. A bacterial mat might cover a patch the size of a room or a field. Without reference, the scene floats free of ordinary proportion. Then a scientist says the depth: 800 meters, 1,500, 2,000, 9,000. The numbers change the image. They pull an invisible column of water into the mind. They add pressure to the frame. They make every animal look more improbable. They make the bubbles seem like messengers crossing a vertical world far larger than the screen. Depth is not just distance down. It is separation from the surface's assumptions. At depth, light has vanished. Temperature has dropped. Pressure has risen. Food from above has thinned. Sound travels differently. Time feels altered. The surface weather may be wild, but the seafloor sees only dim consequences: pressure pulses, currents, sediment movement, occasional falls of organic matter. It is a realm where the ordinary human senses would be nearly useless without machines. And yet, for the animals there, it is not extreme. It is home. We call seeps extreme environments because they are extreme for us and for many organisms. But the organisms that live there are not living in an exception. They are living in conditions to which they are exquisitely fitted. The tube worm does not think of darkness as deprivation. The mussel does not compare methane to sunlight. The bacteria do not need the world to be gentle. They need gradients, reactants, surfaces, and time. This shift matters. It prevents the deep sea from becoming only a theater of human astonishment. The seep is not remarkable because it resembles our world under impossible conditions. It is remarkable because it does not resemble our world and does not need to. At the edge of a seep, ordinary deep-sea life and chemosynthetic life overlap.
A sea cucumber may move across sediment near a mussel bed. A crab may pick through shells. Fish may wait where small prey gather. Anemones may attach to carbonate. Sponges may grow on older rocks. The seep is not a closed kingdom. It has edges, and the edges are lively. Edges are where the chemistry softens. Too close to strong sulfide flow, conditions can be toxic. Too far away, the chemical energy fades. Between those zones, many organisms find opportunity. The community forms rings, patches, and mosaics around invisible gradients. What looks random from a camera may be patterned by molecules. A seep field is rarely one neat vent. It may contain many bubble streams, diffuse flow zones, carbonate slabs, bacterial mats, shell accumulations, live mussel beds, dead mussel beds, tube worm clusters, and bare sediment. Flow may be focused in one place today and another place later. The term field is useful because seepage often spreads across an area rather than emerging from a single point. The seafloor can leak the way old stone leaks groundwater, through cracks, pores, seams, and hidden channels. If hydrothermal vents are sometimes compared to chimneys, cold seeps are closer to damp walls.
But damp walls that feed cities. The comparison is imperfect, but it helps. A seep can be diffuse, creeping, patchy, and still powerful. Its influence is not measured only by the force of a plume, but by persistence. A weak seep that lasts centuries can shape more life than a spectacular burst that ends quickly. In the deep sea, endurance is a kind of intensity. This is one reason carbonate matters so much. It shows endurance. Hard grounds formed at seeps indicate that methane oxidation and mineral precipitation have persisted long enough to alter the seabed. Carbonate is the seep's memory made solid. It also provides rare, hard substrate in muddy environments, allowing corals, sponges, and other sessile organisms to attach. Thus, methane can indirectly create habitat for animals that do not use methane at all. The seep's influence outlives its fuel. A dead seep is not simply dead. It becomes geology for later life. A carbonate outcrop with corals on it may be the afterimage of methane that once moved through that spot. The present community may not reveal the original chemical engine unless someone reads the rock. The seafloor is full of afterimages. Pockmarks are another kind. These are depressions formed by fluid or gas escape from sediments. Some are active, some are relics. From above, in sonar maps, they can look like craters scattered across a slope or shelf. They indicate that the seabed has vented, collapsed, or expelled fluids in the past. But a pockmark does not always mean active methane release today. Some are old scars. Some still leak. Some leak dissolved fluids rather than bubbles. Interpretation requires care. Care is the word that keeps returning. Care in measurement. Care in metaphor. Care in climate claims. Care in exploration. Care in disturbance. The deep ocean punishes overstatement because it is still poorly known. Every confident simplification risks being corrected by the next survey. When researchers found widespread methane seeps on the U.S. Atlantic margin, it was a correction. When Hadal cold seep communities were documented at extraordinary depths, it was a correction. When studies show that much methane from deep seeps is consumed before reaching the air, that too is a correction to simple fear. When shallow Arctic systems raise concern about atmospheric transfer, that is a correction to simple reassurance.
The story moves by corrections. A good voiceover should not hide that. It should let knowledge feel provisional in the best sense. Strong where measured, cautious where uncertain, open where the sea floor has not yet been seen. There is a humility in ocean science that comes from scale. The ocean can always be larger than the survey. The bottom can always be more varied than the map. A seep can always exist beyond the line you ran. In the control room, this humility often appears as silence. The video feed shows something unexpected, and for a few seconds, no one explains it. They look. They let the scene arrive before naming it. This is a good habit. It gives the sea floor a chance to be itself before becoming data. The public rarely sees that part of science. We see the article, the summary, the headline, the discovery. But before that, there is the quiet interval of looking. A bubble stream on a monitor, a patch of white mat, a field of worms at a depth where few expected such abundance. The first response is not always interpretation. Sometimes it is simply recognition that the world has more rooms than expected. The methane seep is one of those rooms. It is a room under pressure, a room without sunlight, a room built by leakage, a room where bacteria make food from chemicals and animals build lives around bacteria. A room connected to the atmosphere by uncertain pathways and to the deep earth by fractures. A room we entered late. If the subject feels philosophical, it is because the physical facts keep pushing toward questions of boundary. What counts as surface? What counts as food? What counts as escape? What counts as waste? What counts as life's source? A seep unsettles these words without needing abstraction. Methane is waste to some organisms, fuel to others, hazard in one context, resource in another, greenhouse gas in the atmosphere, chemical energy at the seafloor, hydrate under pressure, bubble in water, carbon in rock, carbon in tissue. The molecule is not morally one thing.
Its meaning depends on where it is, what it meets, and what happens next. This is true of many things, but methane makes it visible. The ocean floor leaks methane in places where the conditions align, and those alignments are neither entirely rare nor entirely predictable. They are the result of Earth history, ancient plankton, burial, basin formation, plate motion, salt movement, faulting, glaciation, sea level change, ocean temperature, microbial evolution, animal symbiosis, human detection. To say the places is to speak of geography, but also of convergence. A methane seep is a convergence point. Carbon from the surface, heat from below, pressure from water, chemistry from sediment, microbes from the deep biosphere, animals from evolutionary lineages that found a way to live there, technology from human curiosity, climate questions from the present age. All of it meets around bubbles in mud. The title of the subject might suggest a map, and there is a map. Dots along margins, clusters north of Cape Hatteras, plumes along Cascadia, fields in the Gulf, flares around Svalbard, communities in trenches. But the deeper map is not only spatial, it is relational. Each dot connects to processes extending upward, downward, backward, and forward in time. A map of seeps is a map of relationships made visible as locations. Perhaps this is why seep imagery can linger in the mind. It has the strangeness of a secret and the concreteness of a place. The bubbles are real, the animals are real, the chemistry is real. But the implications keep widening until the small scene becomes planetary. There is no need to force awe onto it.
The facts are enough if given room. A methane molecule trapped in hydrate below a continental slope. A microbe using sulfate to oxidize methane in anoxic sediment. A mussel carrying bacteria that turn methane into food. A sonar beam revealing a plume no one knew was there. A trench floor at 9,533 meters supporting tube worms. A fjord in the high Arctic sending gas flares into shallow water. A carbonate rock preserving the memory of a leak after the leak has faded. Each fact has its own atmosphere. Together, they make the ocean floor feel less like an ending and more like a membrane breathing in the dark. The breathing metaphor is tempting, and it should be used carefully. The Earth is not an animal. It does not inhale and exhale with lungs. But metaphors exist because some patterns need felt comparison. Methane seepage is not breathing, yet it shares the quiet rhythm of exchange. Something moves from within to without. Something is transformed along the way. The boundary is alive with process. If we call it breathing, we should remember that the breath is chemical, geological, microbial, and uneven. Not one breath, many small leaks, many fates. In some places, bubbles rise continuously enough to draw a line in the water column. In others, seepage is diffuse, dissolved, almost invisible. In some, methane is trapped in hydrate just below the surface. In others, gas migrates from deeper thermogenic sources. In some, seeps are surrounded by lush animal communities. In others, only microbes mark the flow. In some, the atmospheric relevance may be minimal. In others, especially shallow waters, the sea-air exchange may matter. This variety is the point. There is no single methane seep. There are places. The places where the ocean floor leaks methane. The word places keeps the subject grounded. Not phenomenon, not issue, not resource. Places. Each with depth, sediment, current, organisms, history, and vulnerability. A place can be damaged. A place can be protected. A place can be misunderstood. A place can be revisited. A place can surprise. On a long enough timeline, the seep itself may move. Fluid pathways shift. Sediment seals one crack and opens another. Gas pressure builds elsewhere. A new mat appears. An old mussel bed dies. Carbonate remains. The place is stable only in the loose way a river is stable. Recognizable, but always in motion. This makes conservation difficult. Protecting a seep may require protecting not just the visible community, but the surrounding geological and hydrological system. It may require recognizing that active flow could migrate beyond the exact coordinates first mapped. It may require corridors between habitats. It may require long-term monitoring rather than a single boundary drawn once.
Deep-sea management often struggles with processes that move slowly and invisibly. Methane seeps demand that management think in both geological and ecological time. A ship track is immediate. A tubeworm colony is long-lived. A hydrate layer is climate-sensitive over decades to centuries. A carbonate outcrop may last far longer. The decisions of a permit cycle may affect systems whose natural rhythms do not fit political calendars. Again, the seep teaches scale. It does not teach by speaking. It teaches by being difficult to reduce. Near the end of an ROV dive, the vehicle often lifts from the seafloor slowly. The lights widen, then the details shrink. Mussels become texture. Worms become pale strokes. Bubbles become glitter.
The sediment becomes a gray plain. Then the bottom disappears beneath the vehicle, replaced by black water filled with particles. The ascent begins. For the pilots and scientists, the dive is ending. For the seep, nothing has ended. The moment of human vision closes, but the processes continue. Methane rises, microbes consume, animals feed, carbonate forms. The ocean carries away what dissolves. The deep remains dark. There is comfort in that continuity, but also a kind of ache. We see so little. We leave so quickly. We make meaning from fragments. The seafloor persists beyond our frames. The ending of this story should not resolve too neatly, because the subject itself does not resolve. There is no single conclusion about methane seeps, except that they are more numerous, more varied, more alive, and more consequential than earlier generations imagined. They are places where the planet's hidden carbon emerges into water and becomes part of life, stone, chemistry, and sometimes climate. They are reminders that darkness is not absence, that leakage can build worlds, and that the ocean floor is still full of doors. But even that sounds too final. Better to remain with the image. A cold slope far below the surface. A camera light moving over shells.
A white mat tremoring in the current.
A bubble lifting from a small opening in the mud. It rises, catches the light, and turns silver for a moment before the dark water takes it upward. Below it, the opening remains. Small, persistent, almost nothing. Before that final smallness, there is another way to enter the story. Through sound. A bubble is not silent when it is born. In the deep ocean, as gas squeezes from sediment and detaches into water, it can vibrate. It can make a tiny acoustic event, a pulse too small and too quick for human ears in air, but not too small for hydrophones waiting on the sea floor. Scientists have begun to listen for these signatures because sound can remain when visibility is gone. A camera sees only where its lights are pointed. A hydrophone listens through darkness. In a seep field, the acoustic world may be full of small bursts. Bubble formation, bubble breakup, the low hum of distant ship traffic, the crackle of biological activity, the mechanical noise of instruments. The deep ocean is often called silent, but that is mostly because people do not live in its soundscape. Water carries sound with great efficiency. A seep can be part of that hidden music, marking its activity in frequencies, durations, and repeated clusters. Listening changes the imagination. The ocean floor is not just leaking in light, it is leaking in sound. A hydrophone left near a seep can hear when bubble release strengthens or fades. It can record variability through time, including intervals no ship could remain on station. This matters because methane seepage is not steady like water from a laboratory faucet. It pulses, fluctuates, responds to pressure, pathways, and perhaps disturbances. A few hours of video may show beauty. Weeks of sound may show behavior. There is a discipline in leaving instruments behind. Human attention wants the vivid moment, the dive, the image, the creature crossing the frame. But many truths of the seafloor appear only when no one is watching. They appear in long records, in numbers changing slowly, in a bubble counter registering bursts through the night, in a chemical sensor showing dissolved methane rise after a current shift, in a lander camera capturing the same patch of mud again and again until change becomes visible.
At seep sites near the edge of hydrate stability,
This long watching becomes even more important. Scientists want to know not only that methane emerges, but why, when, and how much. Is the gas coming from hydrate breaking down? From deeper free gas? From microbial production? From a mixture? Does the seep respond to bottom water temperature? Is it controlled by subsurface pressure? Does the bubble flux change after storms or earthquakes? Does microbial oxidation keep pace with emission? These questions cannot be answered with a single dramatic image. They require patience equal to the seep's own patience. The instruments placed on the seafloor are not glamorous in the way animals are. Metal frames, glass spheres, batteries, sensors, cables, acoustic heads, sampling ports. Yet they become a temporary extension of human perception. For a season or a year, they sit where people cannot, enduring cold, pressure, sediment, currents, and darkness. They do not understand what they record, but they remember. When they are recovered, the shipboard deck becomes a place of suspense. A lander breaks the surface, wet and heavy, carrying months of unseen time. The data are inside. Perhaps the batteries lasted. Perhaps the sensors drifted. Perhaps the instrument was tilted by currents or half buried by sediment. Perhaps it captured exactly the pattern scientists hoped to see. A hard drive can become a memory of the seafloor. The story of methane seeps is therefore also a story of how humans extend senses into the deep. We build machines that see, listen, smell chemically, feel pressure, measure temperature, count bubbles, and return with fragments. Our bodies are weak down there, but our tools become patient. Still, tools shape what is noticed. A sonar survey finds bubble plumes, not necessarily diffuse seepage. A camera sees large animals and mats, not the full microbial community. A core samples one narrow cylinder of sediment, not the whole chemical field. A seismic profile images subsurface structure, not the living surface. Each method brings a kind of blindness. Only by combining them does the seep begin to appear as more than a partial truth.
The ocean reminds researchers to be plural. No single instrument owns the deep. In the Gulf of Mexico, plural methods have revealed one of the most complex seep provinces on Earth. The region is a layered archive of salt movement, hydrocarbon migration, brine flow, carbonate formation, gas hydrate, oil seepage, and animal symbiosis. Some sites look almost theatrical under the lights. A dark brine pool with a sharp surface, a ring of mussels along the rim, tube worms standing nearby, oily droplets moving with a slow, strange grace, and white bacterial mats marking chemical seepage. Yet the theater is not staged. It is geology acting through fluid. Salt is central there. Ancient evaporite deposits under the Gulf behave like a buried, mobile foundation. Because salt is less dense than the overlying sediment and flows under pressure, it rises in domes, sheets, and ridges. It fractures the layers above, creates traps for hydrocarbons, redirects fluids, and shapes the seabed. Methane seeps in this setting are not isolated oddities. They are expressions of a basin whose deep structure has been moving for millions of years. The brine pools may be the most visually haunting. Their salinity is so high that the dense brine collects in depressions instead of mixing easily with seawater. A submersible can hover over what appears to be a shoreline, complete with a reflective interface. The illusion is powerful, a lake under the ocean. But the water in that lake is chemically hostile to many animals. It can be low in oxygen, rich in methane, and loaded with sulfide.
Along its margins, however, life uses the very danger that excludes others. A mussel at the rim of a brine pool lives at a boundary of extremes. Too far from the brine, the chemistry that feeds its symbionts may weaken. Too far into it, the animal may encounter lethal conditions. It must occupy the edge. It must live precisely where gradients are steep. This is not a poetic exaggeration, it is ecology. The animal's place is determined by molecules moving through water.
This edge living is common at seeps. The most productive zone is often not the pure source, but the meeting point between source fluid and seawater. Chemical energy requires contrast. Methane, sulfide, oxygen, sulfate, nitrate, and other compounds must meet in the right arrangements for microbes to harvest energy. Life gathers where difference is usable. On land, we often imagine stability as sameness. At seeps, life depends on controlled difference. The same principle operates in hydrothermal vents, but the conditions differ. Vents release hot, often acidic, mineral-rich fluids driven by volcanic heat. Seeps release cooler, hydrocarbon-rich fluids driven by sedimentary, tectonic, or hydrate-related processes. Vent communities can be destroyed and reborn with volcanic activity. Seep communities can persist for longer periods, though they too shift with flow. Both show chemosynthesis, but their tempos and architectures differ. Understanding the difference prevents a common confusion. Not every deep-sea oasis is a vent. Not every chemical ecosystem is hot. Some of the most extraordinary deep-sea communities are built around cold leakage, not fire. Cold does not mean inactive. Cold can feed. Cold can build. Cold can leak for centuries. In the northwestern Atlantic, cold seeps have revealed another dimension of surprise. Before widespread mapping, parts of the margin were not expected to contain extensive methane seepage. The geological setting seemed comparatively quiet. Yet, sonar data from research vessels showed plumes rising from the seafloor, and ROV dives confirmed habitats with bacterial mats, mussels, anemones, quillworms, hydrate, and carbonate. The margin became a reminder that absence of expectation is not evidence of absence. The updated inventory of Atlantic margin seeps more than doubled the known sites. When probable duplicates were removed, researchers identified about 1,139 unique gas flare sites and about 275 distinct seep locations or clusters across the region. Most were concentrated north of Cape Hatteras, with many in relatively shallow upper slope depths. This kind of numerical knowledge can sound dry, but behind it is a transformed map. A coast once thought to have few seeps now has a constellation. Numbers are another kind of imagery. 1,000 flares along a margin, hundreds of clusters, 47 clusters with multiple seeps, depth bands where seeps concentrate, and other depth ranges where they do not. Each number hints at unseen terrain, at water columns crossed by bubbles, at slopes where chemical energy reaches the surface. But numbers cannot replace the dive. They cannot show the soft trembling of a bacterial mat, or the way mussels stack over carbonate, or the faintness of bubbles in ROV light. They cannot show the moment when a scientist realizes a sonar target is not just acoustic backscatter, but a living seep ecosystem. The map and the image need each other. Without the map, the image is isolated. Without the image, the map is abstract. The Atlantic seeps also reveal the importance of canyons. Shelf break canyons cut into the continental slope, exposing strata, focusing sediment transport, altering fluid pathways, and shaping hydrate stability.
Some seeps occur on promontories near canyon heads, where the three-dimensional geometry of the seafloor and hydrate stability zone may create conditions favorable for fluid escape. The seafloor's shape is not just scenery, it is plumbing. A canyon can be a wound in the slope, or a corridor, or a window into buried layers, or a place where currents, sediments, and fluids interact in ways that make methane escape more likely. Deep sea geography is active. A ridge, canyon, scarp, pockmark, or mound is not merely a form. It affects pressure, flow, erosion, deposition, and habitat. The animals live on a landscape shaped by processes they cannot see, but depend on. In the Arctic, the landscape includes ice history. Glaciations loaded and unloaded shelves. Sea level rose and fell. Permafrost formed on exposed shelves during lower sea levels and was later submerged. Hydrate stability shifted with pressure and temperature. Organic-rich rocks and sediments generated gas. Fjords carved by glaciers became sites where faults, fractures, and sediment pathways could deliver methane to shallow water.
The Arctic is not a single methane story. It is many. Some gas flares in Svalbard fjords appear linked to bedrock source formations and fault-controlled migration. Along the West Spitsbergen margin, gas bubble plumes have been associated with the landward limit of hydrate stability and warming Atlantic water, though the degree to which methane reaches the atmosphere may be limited by dissolution and microbial oxidation in many settings. On broad Siberian shelves, subsea permafrost and shallow methane emissions raise different questions. In the Beaufort Sea, ancient carbon sources and microbial filters complicate the path from sediment to air. The public often wants one Arctic methane answer. The Arctic refuses. It offers shallow shelves, deep slopes, fjords, permafrost, hydrate, faults, old seepage, new warming, strong seasonal cycles, sea ice change, and measurement challenges. It is both a warning region and a region where overstatement can travel faster than data. The correct emotional response is alertness, not simplification. Methane seeps in shallow Arctic waters matter because the water column may not fully consume the gas before it reaches the surface. They matter because warming is rapid. They matter because frozen carbon stores can be sensitive. They matter because local emissions may affect regional carbon budgets. But they must be interpreted with careful source tracing and long-term monitoring. A bubble in an Arctic fjord is not automatically proof of a new climate feedback. It is a clue that must be placed into geology, history, and seasonal context. This care does not weaken concern. It strengthens it. A concern grounded in real mechanisms is more durable than fear built from a single image. The Arctic deserves close attention precisely because its systems are complex, changing, and unevenly measured. The Hadal Trenches, by contrast, bring the story to almost unimaginable depth. The discovery of extensive chemosynthetic communities in the Kuril-Kamchatka and Western Aleutian Trenches extends the known realm of seep life downward. There, the cold seep model intersects with subduction geometry and trench sedimentation. Organic matter trapped in deep V-shaped basins can be converted into methane under anoxic conditions. Compression and faulting associated with plate bending and subduction can guide fluids upward. At the sea floor, methane supports communities where sunlight has no presence and pressure is crushing. It is tempting to call these places alien. But alien is often a lazy word for earthly things we have not yet accepted. They are not alien.
They are part of Earth. They are Earth at full depth. They are as native to the planet as forests, rivers, reefs, deserts, and glaciers. Calling them alien may capture a feeling, but it risks placing them outside responsibility. Better to say they are unfamiliar. Better to say they expand what familiar should mean. A tube worm field at 9,000 meters is not a fantasy. It is a biological fact. It tells us that Earth's biosphere extends into pressure regimes that few machines can enter. It tells us that chemical energy can support complex communities in the deepest trenches. It tells us that our mental picture of the Hadal Zone has been too sparse. And because the discovery is recent, it also tells us how young our knowledge remains. Humanity has mapped planets and moons from afar, but our own Hadal trenches still conceal ecosystems. This is not because humans are foolish. It is because depth is hard. But the result is the same. The ocean floor remains a frontier, not in the romantic sense of emptiness, but in the scientific sense of incomplete encounter. The word frontier can be dangerous too. It can imply conquest. The deep sea does not need conquering. It needs understanding, and in some places, restraint. A frontier of knowledge does not have to become a frontier of extraction. The seep story sits in this tension. Methane hydrates are potential energy resources. The deep sea contains minerals. Continental margins host oil and gas. The technologies that explore also enable access. Every new understanding of seabed processes can be turned toward both protection and use. The future of methane seep habitats may depend on which impulse dominates. A long-form story can slow the impulse to use before understanding. It can make the place feel real enough to matter. Because much of environmental harm begins with abstraction. A habitat becomes a lease block. A seep becomes a feature. A community becomes benthic fauna. Methane becomes flux. Carbon becomes units. These terms are necessary for science and management, but something is lost if they are the only language. The mussel bed must remain a mussel bed. The bubble must remain a bubble. The seep must remain a place.
This does not mean abandoning precision. It means precision with texture. A cold seep is a location where hydrocarbon-rich fluids, often methane and hydrogen sulfide, emerge from below the seafloor. That is precise, but it is not enough. The same place may be a microbial factory, an animal habitat, a carbonate builder, a climate node, a geological signal, and a vulnerability. Precision should open the world, not shrink it. The deeper one goes into methane seeps, the more interdependence appears. Methane production depends on buried organic matter. Buried organic matter depends on surface productivity and sedimentation. Fluid migration depends on geology. Microbial oxidation depends on electron acceptors and community structure. Animal communities depend on microbial symbioses and habitat stability. Carbonate formation depends on chemistry altered by microbes. Climate relevance depends on methane's path through sediment, ocean, and atmosphere. Human response depends on knowledge, values, and choices. No part stands alone. This interdependence is why the seep can feel like a miniature Earth system. It contains the same complexity as larger planetary cycles, concentrated into a visible patch of seafloor. The coldness of seeps also matters emotionally. Hydrothermal vents are spectacular in a volcanic way.
Black smokers billow mineral-rich fluid like underwater chimneys from another age. Cold seeps are subtler. Their drama is quieter. A white mat, a shell bed, a bubble stream, a brine edge. They ask for a slower gaze. They do not overwhelm with heat and smoke. They invite attention to small signs. This makes them well-suited to long-form storytelling. They unfold. A viewer sees mud, then bubbles, then mats, then animals, then chemistry, then geology, then climate, then the deep time behind it all. The subject expands without needing to shout. The mystery is not what exploded. The mystery is what has been quietly happening all along. There is also a contradiction at the heart of the seep. Methane is both a sign of decay and a source of life. It often begins with the breakdown of organic matter, the afterlife of dead plankton and buried carbon. Yet, at the seep, that breakdown becomes energy for new communities. Death feeds microbes. Microbes feed animals. Animals build habitats. The line between decay and creation becomes difficult to draw.
The seafloor is good at recycling meanings. A dead diatom becomes methane. Methane becomes bacterial biomass. Bacteria become mussel nutrition. Mussel shells become carbonate-bound habitat. Carbonate becomes coral substrate. The original sunlight that fed the diatom may be long gone, but its carbon keeps changing form. At seeps, the carbon cycle is not an abstraction, but a transformation sequence. One could tell the entire story through the life of that carbon. Born in light, buried in darkness, converted by microbes, held in hydrate, released through sediment, consumed again by microbes, built into an animal, locked into stone. Perhaps eventually subducted, buried, heated, and transformed again. The carbon does not rest. Even when buried, it is waiting inside processes. This restless carbon is part of why the planet's history is so dynamic. Carbon moves between atmosphere, ocean, rocks, sediments, mantle, and life. Over geological timescales, these movements regulate climate, support ecosystems, and alter the chemistry of sea and air. Methane seeps are one set of valves in this immense system. Not the only valves, not always the largest, but visible, measurable, biologically rich, and emotionally powerful. The valve metaphor is useful but incomplete because valves imply engineering. Natural seepage is messier. It is not controlled by a single handle. It is controlled by sediment permeability, fractures, gas pressure, hydrate stability, microbial consumption, and regional geology. It can clog, migrate, pulse, and bypass. The seafloor is plumbing made by accident and time. Perhaps plumbing is too ordinary for something so strange, but ordinary words can help. The deep earth has pipes, seals, reservoirs, leaks, traps, and pressure gradients. Petroleum geologists know this language well. Marine biologists bring another language: symbiosis, succession, colonization, trophic webs. Microbiologists bring another: metabolic pathways, archaea, sulfate reducers, methanotrophs. Oceanographers bring currents, stratification, dissolved gases. Climate scientists bring fluxes, budgets, feedbacks. A methane seep requires translation among these languages. That translation is part of the intellectual beauty of the subject. No single discipline can own it. A geologist may explain why methane arrives. A microbiologist explains what consumes it. A biologist explains who lives on it. An oceanographer explains where it goes. A climate scientist explains what it may mean beyond the seep. Together, they build a fuller story. But even together, they are still building. In many seep ecosystems, species remain undescribed. Microbial interactions remain unresolved. Flux estimates remain uncertain. Hydrate responses to warming remain debated in detail. Long-term records are sparse. The deep sea keeps its advantage. This should not discourage us. It should change the tone. The correct tone is not final mastery, but attentive wonder. Not ignorance, but unfinished knowledge. A slow documentary voice belongs here, because the subject itself resists hurry. Methane seepage is not a quick headline. It is a long exchange between buried carbon and ocean water. It deserves sentences that can travel. It deserves quiet after facts. It deserves images that remain physical, not inflated into empty grandeur. Look again at the bubbles. They are small. They rise. They may vanish before reaching the surface. They may not change the climate directly. They may still feed an entire hidden community. Importance is not always proportional to spectacle. Some of the most consequential processes on Earth occur without spectacle. Microbial metabolism, sediment burial, gas dissolution, carbonate precipitation, ocean circulation, heat diffusion. Methane seeps bring some of these processes close enough to see, but they do not make them loud. They remain subtle even when illuminated. The danger in telling this story is to make every sentence profound. The seafloor is not profound all the time. Sometimes it is just mud. Sometimes a fish swims through the frame and nothing happens. Sometimes a sample fails. Sometimes a bubble stream is small. Sometimes a mat is patchy. The quiet, ordinary details keep the wonder honest. A crab crosses the shell bed. A worm retracts. A sensor blinks. Sediment falls from the manipulator claw. The water clouds. The pilot waits. The seep continues. This is the rhythm of actual exploration. Long intervals of searching, sudden recognition, careful sampling, technical problems, long ascents, data processing, return, revision. The final scientific picture is built from patience more than revelation. And patience may be the hardest thing to bring to climate-related subjects. The modern world is trained to expect catastrophe or reassurance, alarm or dismissal. Methane seeps do not fit that tempo. They ask for sustained attention to processes that can be slow and still matter, natural and still affected by change, buffered and still uncertain, remote and still connected. The ocean floor leaks methane. The sentence is true. But after hours inside the story, it should no longer sound simple. It should carry the Atlantic margin, Cascadia, the Gulf, Svalbard, the trenches, the microbial mats, the hydrate cages, the sonar flares, the brine pools, the tube worms, the oxidation filters, the carbonate records, the climate questions, the instruments listening in darkness, and the unresolved map. It should sound like a planet with seams. The ending, when it comes, should not close those seams. It should leave them open, because they are open. That is the point. The seabed is not a perfect seal. The ocean is not a passive cover. Life is not confined to the sunlit logic we first learned. Carbon does not stay in one form. The deep is not empty. Somewhere, now, while storms move over the surface and satellites cross the night side of Earth, methane is entering seawater from the floor of the ocean.
It is happening along a slope beyond the continental shelf. It is happening in a basin shaped by salt. It is happening near a fault. It is happening in a fjord. It is happening, perhaps, in a trench where no camera is present. Some of it is being eaten before it can rise. Some is dissolving. Some is feeding microbes inside animals. Some is becoming stone.
No headline marks it. No horizon shows it. The process is too dispersed, too quiet, too deep. But it is part of the planet's present tense. To think about methane seeps is to think about what continues without us. It is to let the mind descend below the familiar ocean, below waves, below fisheries, below cables and charts, below the blue surface that has carried so much human imagination, into a place where the earth leaks chemistry through its own floor and life gathers around the leak. There is nothing sentimental about that gathering. It is survival. It is metabolism. It is evolution using what is available. It is a mussel opening in cold water. It is a bacterium dividing in sediment. It is a tube worm holding its plume into the current. It is a bubble rising toward a long, dark column it may never cross. And perhaps that is enough for awe. Not the claim that the seep explains everything, not the claim that it saves or dooms the world. Only the fact that in places almost no one will ever see directly, beneath pressure and darkness, the boundary between earth and ocean is alive with exchange. The surface of the planet is not where the story ends. It continues under the mud. There is one more scale to consider, the scale of a human lifetime beside a seep animal. Some seep tube worms are thought to live extraordinarily long lives. They grow slowly in cold, energy-limited environments, extending their tubes over years and decades, sometimes longer than the careers of the scientists who study them. This longevity changes the emotional weight of a field of worms. They are not seasonal grass. They are old structures made of living tissue, microbial partnership, and accumulated time. A vehicle can cross them in minutes, but their growth may span centuries. A mussel bed also holds time, though differently. Shells accumulate. Living mussels sit atop dead ones. The bed thickens into a rough, layered archive of recruitment, mortality, and chemical persistence. In the spaces between shells, smaller animals find refuge. Sediment collects, bacteria grow, predators hunt. The mussel bed becomes habitat within habitat, not merely a population, but a three-dimensional structure. This architecture is easily underestimated. From a distance, a mussel bed may look like debris. Up close, it is a city of surfaces. Each shell changes current flow. Each gap shelters something. Each living mussel filters water and maintains symbionts. Each dead shell remains as structure. The bed is both community and monument. At older seep sites, dead shells can be so abundant that they become part of the geological signature. Shell hash, carbonate, tube remains, and chemical minerals combine into a record of former flow. A scientist can collect a piece of rock or shell and bring back evidence of seepage long after the active bubbles have moved elsewhere. In this way, seeps write themselves into the seafloor with biological handwriting. The writing is not always easy to read. A carbonate slab might indicate methane oxidation, but its age, formation conditions, and relation to present flow require analysis. A shell bed might be active, relict, transported, or mixed. A pockmark might be ancient or currently seeping. A sonar flare might be methane, fish, or another acoustic scatterer until confirmed. The ocean offers clues, not labels.
This uncertainty is not a weakness of the science, it is the nature of the subject. The seep is a process crossing categories, and processes often leave ambiguous traces. Good science becomes the art of reducing ambiguity without pretending it never existed. In laboratories after expeditions, the seep is taken apart in fragments. A vial of water is analyzed for methane concentration. A sediment core is sliced by depth. Pore water is extracted. Sulfate, sulfide, alkalinity, chloride, dissolved inorganic carbon, and isotopic ratios are measured. DNA is sequenced. Shells are identified. Carbonates are dated. Hydrate samples, if recovered, are kept under pressure or studied before they dissociate. The vivid seafloor becomes columns, curves, chromatograms, microscope images, and tables. There is a different beauty in this transformation. The bubble seen on video becomes a point in a dataset. The white mat becomes microbial community composition. The carbonate ledge becomes mineralogy and isotopes. The mussel becomes host symbiont genetics. The place becomes many forms of evidence.
Yet the lab can never fully replace the place. The sample is a translation, and every translation loses something. Pressure changes, temperature changes. Microbial rates may shift when brought to the surface. Hydrate may dissociate. Animal behavior cannot be fully understood in a jar. This is why in situ measurement is so valuable. The deeper the environment, the more the act of recovery can alter what is being studied. The deep sea is not easily brought up. It has to be met where it is. That is why some instruments are designed to preserve pressure, to sample gases without losing them, to incubate microbes at depth, to measure chemistry directly in the water column, to observe bubbles as they rise rather than infer their history afterward. Each improvement is a way of respecting the environment's conditions. The more faithfully scientists can keep the deep sea deep, the better they can understand it. Methane seeps have also changed how we think about the deep biosphere. Beneath the seafloor, microbial life extends through sediments and rocks far below the visible surface. Some of this life is slow beyond ordinary imagination, with metabolic rates so low that cells may persist over immense timescales. Methane production and consumption are part of this hidden biosphere. A seep is where that buried microbial world becomes connected to the larger ocean. The animal communities are dramatic, but the microbial world is the deeper foundation. Without microbes, methane would not be produced in the same way. Without microbes, much methane would not be consumed in sediment. Without microbes, sulfide would not be generated and oxidized into food for symbiotic systems. Without microbes, carbonate precipitation would be different. The animals are the visible crown of a microbial process. This inversion is important. Humans naturally focus on animals because animals are closer to our scale. We see mussels, worms, crabs, and fish, but the seep's primary engines are microscopic. The deep ocean teaches a lesson repeated across Earth. The small rules the large more often than human intuition expects. A bacterial mat may look like a stain. It is an ecosystem engineer. A consortium of archaea and bacteria may live in an invisible sediment layer. It may determine how much methane escapes. A symbiont inside a mussel's gill may produce the carbon that builds the mussel's body. Scale does not equal importance. The methane seep is a democracy of sizes, though not an equal one. Molecules, microbes, animals, rocks, currents, and plates all exert power at their own scales. The system exists because none of them acts alone. There are also chemical details that complicate the phrase methane seep. Many cold seeps release more than methane.
hydrogen sulfide, heavier hydrocarbons, oil, brine, carbon dioxide, and other reduced compounds may be present. Some animal symbioses depend more on sulfide than methane. Some microbes oxidize methane directly, while others use sulfide produced by methane oxidation. Thus, the ecosystem can be methane-based in origin, but sulfide-based in immediate energy for some organisms. A tube worm, for example, may not consume methane directly. Its symbionts may rely on sulfide. But that sulfide can be linked to methane oxidation in the sediment. The methane fuels a reaction that produces sulfide, and sulfide fuels bacteria that feed the worm. The path is indirect, but essential. This is another reason simple labels can mislead. A methane seep is often a methane sulfide carbonate system with multiple chemical loops. The animals may be arranged around whichever compounds their symbionts use. A mussel species with methane oxidizing symbionts may occupy different microhabitats than one relying on sulfide oxidizers. Some hosts carry both types. Evolution has allowed flexibility in certain lineages, and that flexibility can determine where they thrive. The more precise the science becomes, the more intricate the story becomes. And yet, the viewer does not need every reaction equation to feel the mystery. The essential idea is physical. Chemicals from beneath the seafloor feed microbes, and microbes make life possible in darkness. The details deepen the idea without replacing it. A long-form narration can move between these levels. It can show a bubble, then explain hydrate, then return to the mussel shell, then widen to climate, then descend into the sediment pore. This movement mirrors the seep itself, where scales are connected vertically. The story should not feel like a list because the system is not a list. It is a flow. Flow is the most important word. Fluids flow through sediment. Methane flows through pore spaces. Electrons flow through metabolism. Carbon flows through organisms. Water flows through the ocean. Energy flows through communities. Information flows from instruments to ships to labs to maps. The seep is flow made local. When flow stops, the community changes. Microbial mats may fade. Mussels may die. Tube worms may disappear. Carbonate remains. Other animals arrive. The habitat shifts from chemosynthetic to suspension feeding or background benthic life. The story does not end. It changes energy source. This shift can be seen as ecological succession, but not the familiar forest succession of grasses, shrubs, and trees. It is succession governed by chemical seepage. Microbes colonize new seepage. Carbonate forms. Symbiotic animals establish. Flow declines or migrates. Non-chemosynthetic organisms colonize the hard substrate. The seep's legacy supports a new community. A place built by methane may eventually be inhabited by creatures that do not use methane. There is poetry in that, but it is also practical ecology. Hard substrate is valuable in the deep sea, especially in soft sediment regions. Carbonate formed at seeps can become islands for corals and sponges. Thus, methane influences biodiversity in ways not limited to active seep organisms. This afterlife complicates impact assessment. If an area contains inactive seep carbonates with coral communities, it may still owe its existence to past seepage. Protecting only active bubble streams could miss the broader habitat mosaic. A seep system includes active, waning, relict, and successor habitats. The boundary is temporal as much as spatial.
The ocean floor is a palimpsest. Old seepage under new life. New seepage through old rock.
Ancient carbon in modern microbes. Modern warming touching ancient hydrate. The layers overlap. Human stories often prefer beginnings and endings. A seep offers phases instead. Birth is not a single moment. Death is not absolute. Activity migrates. Communities turn over. Rock remembers. Carbon keeps moving. This phase-like quality is also true for methane's climate role. The molecule may pass through multiple transformations before it has any atmospheric relevance. Its warming potential matters only if it reaches the atmosphere as methane. Before that, it can be oxidized, dissolved, stored, or incorporated. The path determines the effect. Climate models therefore need not only stores of methane, but rates of release, oxidation, transport, and exchange. This is hard to measure because the ocean is dynamic. Currents can carry dissolved methane away from a seep plume, spreading it laterally before oxidation is complete. Water masses move. Stratification can trap or mix. Microbial communities respond to methane availability. Oxygen can become locally depleted if oxidation is intense. Carbon dioxide produced by oxidation can alter local carbonate chemistry. A seep is a source, but the ocean determines the plume's fate. In shallow water, wind and waves may transfer gases across the surface more directly. In ice-covered regions, sea ice can block, store, or seasonally release gases. In fjords, stratification and circulation can create unique conditions. In deep water, pressure and distance favor dissolution and microbial consumption. There is no universal fate. This is why the phrase "methane from the ocean floor" should always be followed by a question: where? Depth is destiny for a bubble. Geology is destiny for a seep. Microbiology is destiny for a molecule. Climate relevance is destiny after all those destinies have acted. Some of the most dramatic public images of methane seepage show bubbles reaching the sea surface or even flames lit over methane-rich water. These images are real in some contexts, especially shallow settings, but they do not represent all seeps. Deep methane seepage often remains a deep ocean process. The ocean is not transparent to methane in the way a chimney is transparent to smoke. It reacts. This distinction matters because fear can travel faster than accuracy. A story that shows methane bubbles in the deep and implies direct atmospheric escape from every plume would be misleading. A story that dismisses all seep methane because many deep plumes are consumed would also be misleading. The honest story must keep the pathway visible. Perhaps the most useful image is not a chimney, but a maze. Methane enters the maze from below. It may be consumed in sediment, trapped in hydrate, released as bubbles, dissolved into water, eaten by microbes, converted into carbon dioxide, transported by currents, incorporated into biomass, precipitated as carbonate, or, in some settings, reach the air. The maze has many exits, and most molecules do not take the same path. The places where the ocean floor leaks methane are entrances to that maze. The scientific task is to follow the molecule without losing it in metaphor. A narrative task is different but related, to let the audience feel the maze without becoming lost. The way to do that is through scenes. A ship over the Atlantic margin, a sonar plume on a screen, a Gulf brine pool like a dark lake, Cascadia bubbles along a subduction slope, Svalbard Fjord flares in shallow Arctic water, Hadal tube worms in a trench, a lab vial of water, a carbonate rock in a gloved hand. A muscle opened carefully to reveal tissue full of symbionts. A hydrophone listening to bubbles. Each scene holds one aspect of the system. Together, they create a mental map. The strongest scene may still be the quietest, a bubble stream rising beside a mussel bed. It contains source, pathway, ecosystem, and uncertainty. We do not know by looking how much methane is in the plume, how much will dissolve, how much has already been eaten below, how old the seep is, how long the mussels have lived there, whether the flow will continue next year, or whether the carbonate under the shells formed over centuries. The image is both evidence and question.
Good science begins there, not with certainty, but with a disciplined response to mystery. The reference style you asked for leans into large mysteries and slow unfolding, and methane seeps suit that rhythm because they are mysteries with instruments pointed at them. They are not supernatural, they are measurable. But measurement does not flatten them. It reveals deeper strangeness. A methane seep is one of those subjects where the explanation increases the wonder. Knowing that microbes consume methane anaerobically does not make the white mats less eerie. It makes them more alive. Knowing that mussels host bacteria does not make the mussel bed less beautiful. It makes every shell a collaboration. Knowing that hydrate exists under pressure does not make fire ice less strange. It makes the seafloor's temperature-pressure world more tangible. Knowing that deep methane often fails to reach the atmosphere does not make the bubble plume less important. It places it in a larger system of sinks and transformations. Knowing that Hadal trench communities are supported by methane-rich fluids does not make them less astonishing. It moves astonishment from vague amazement to grounded respect. The ocean floor leaks methane, and the leak becomes a lens. Through it, we see the deep biosphere. Through it, we see the carbon cycle. Through it, we see the limits of our maps. Through it, we see life's ability to use hidden energy. Through it, we see the difference between a climate threat and a climate pathway. Through it, we see how much of Earth remains active beyond ordinary sight. There is a scene from many oceanographic cruises that never appears in final footage: the hours of nothing. The ship runs survey lines. Screens show bathymetry slowly filling in. People rotate watches. Someone eats a late meal from a plastic tray. The sea outside is black. The instruments ping. The data accumulate. Most of the time, discovery feels like waiting. Then, the water column display shows a flare, a vertical streak where no streak was expected. The room sharpens. Coordinates are marked. The line is checked. Was it noise? Was it fish? Is it repeatable? Another pass is made. The flare appears again. The map gains a target. The target becomes a dive plan. The dive plan becomes descent. The descent becomes seafloor. The seafloor becomes bubbles. This chain from anomaly to place is one of the quiet dramas of modern exploration. The ocean does not reveal itself all at once. It hints. Scientists follow hints with expensive caution. The first hint may be sound returning from bubbles. The final confirmation may be a living community around a seep. Between them lies the human craft of inference. In a world saturated with instant images, this slow inference is almost old-fashioned. It asks people to care about incomplete evidence, to revise expectations, to tolerate uncertainty, and to wait for confirmation. Methane seep science is therefore not only a set of facts about the ocean, it is a way of knowing a hidden planet. The hidden planet is not elsewhere. It is under the blue surface.
a surface that covers most of Earth. This fact alone should make humility easy, but it often does not. We live on land, measure from land, govern from land, and imagine the ocean mostly where it touches land. The deep sea floor is out of sight, so it becomes background. Methane seeps resist that backgrounding. They show that the floor of the ocean has active chemistry, dense habitats, and climate connections. They make the background speak. The speaking is soft, a sonar flare, a bubble sound, a microbial isotope, a carbonate crust, a worm field. If we do not listen carefully, we miss it. The ending of the story can therefore be understood not as a conclusion, but as a listening posture. The ocean floor continues its quiet emissions. Some are ancient, some are changing, some are mapped, many are not. The task is not to solve the seep once and for all, but to keep listening well enough that future changes can be understood.
Listening well means baselines. Without baseline data, change is difficult to detect. If a seep strengthens, weakens, migrates, or shifts community composition, scientists need earlier records for comparison. This is especially important in a warming ocean. A baseline is not glamorous, but it is one of the most important gifts science can give the future. A record of what was true before the next change. The deep sea has too few baselines. Many places will change before we have watched them long enough to know how unusual the change is. This is a quiet tragedy of late discovery. We are learning the deep ocean at the same time we are altering the planet. Some systems may be disturbed before they are described. Some signals may be misread because the past was never measured. Methane seeps, with their climate relevance and habitat uniqueness, sit directly inside this problem. But the situation is not hopeless. Every expedition adds knowledge. Every mapped seep improves the baseline. Every long-term instrument helps separate natural variability from trend. Every protected area that includes seep habitats preserves living laboratories. Every careful public story can make the deep sea less abstract. Understanding is not the same as saving, but it is often a prerequisite. The final emotional movement should be toward that kind of quiet responsibility, not a command, not a slogan. A sense that the deep ocean is part of the inhabited world, even where humans do not live. If a methane seep could be held in the mind properly, it might change the shape of environmental imagination. It would show that beauty is not always colorful, that importance is not always visible, that climate processes can be local and molecular, that life can flourish around toxicity, that geological time and biological time overlap, and that a leak can be a habitat. It would also show that mystery does not disappear when named. Cold seep, methane hydrate, anaerobic oxidation, chemosynthesis, Pathimodiolus, Siboglinid, carbonate, pockmark, gas flare. Each name makes the place more knowable, but the whole remains larger than the names. This is the best kind of science, not the end of wonder, but wonder with handles. At the end, after all the names, there is still the dark water. There is still the small silver bubble. There is still the animal opening its shell in a current made useful by bacteria. There is still the carbon that once drifted in sunlight and now rises from below the sea floor. There is still the pressure of an ocean resting over a leak. There is still the vastness above the frame. The seep does not need to be made symbolic. It is already itself, and itself is enough.
a place where the ocean floor is not closed, where methane escapes and is transformed, where life gathers without sunlight, where the planet's hidden carbon briefly becomes visible before disappearing again into water, rock, bodies, and time. One last descent can hold the whole story. Imagine the vehicle falling through the water at night, although night is only a surface word. Above, the ship rolls softly. Below, the ROV passes through layers of temperature and pressure. The cable behind it hangs like a black thread connecting human curiosity to a place no human body could enter alone. Particles drift past the camera in the lights, each one a small piece of the upper world falling into the lower one. For a while, there is no bottom, only water. Then the altimeter begins to count differently. The pilots adjust the descent. A pale floor forms out of darkness, first as haze, then as texture. Mud, ripples, a small fish turning away. The vehicle slows, levels out, and begins to fly a few meters above the sediment. At first, nothing suggests methane. This is important. The extraordinary does not always announce itself. The sea floor may appear empty for long stretches, and emptiness is its own ecosystem. But the human eye searches for signs. A white patch, a shadow of carbonate, a glint of shell, a tremor in the water. The pilots follow a slope. The scientists compare the view with the sonar target. Everyone knows the plume should be near, but the bottom does not care where people expected the answer.
Then the lights catch a faint vertical shimmer. A bubble rises. It is almost too small for the amount of meaning that will be placed upon it. The vehicle moves closer. More bubbles appear. They come from a point in the sediment, then another point a few meters away. The mud around them is pale with bacteria. Shells lie half-buried. Some are dead, some alive. A crab pauses at the edge of the light. The scene is not grand, but it is unmistakable. The ocean floor is leaking. The first response is practical. Record position, note depth, zoom in, check for hydrate, look for fauna, sample the gas if possible. Take water above the plume, push core through the mat, avoid disturbing the mussel bed, watch the thruster wash, keep the vehicle stable. This practical language is part of the beauty. It keeps wonder from becoming vague. Awe does not prevent careful work, it requires it. The seafloor deserves the steadiness of people who can be moved by what they see and still remember to measure it properly. The manipulator reaches down. Its metal fingers move with surprising delicacy. A core tube presses into the sediment. Bubbles slip past. When the tube is lifted, it carries mud, water, microbes, chemistry, and perhaps a record of methane oxidation layered by depth. In the lab, that plain cylinder may reveal where sulfate disappears, where methane rises, where carbon changes isotope, where microbes cluster, where the invisible engine runs. A second sample collects mussels. This is always a more uneasy moment. To understand the animal, one must sometimes remove it. The camera watches shells close as the manipulator lifts them. The sample box becomes a small prison of deep-sea life on its way to pressure change, cold rooms, genetic analysis, microscopy, and human knowledge. Science is careful, but it is not without cost. Every sample is a negotiation. Good researchers know this. They sample enough to understand, not enough to consume the place. They photograph widely, take only what is needed, preserve context, and return data to a broader purpose.
The deep sea has already suffered from the assumption that what is far away is abundant beyond consequence. Seep science, at its best, works against that assumption.
The ROV continues. The bubble streams become more numerous.
The sea floor rises slightly into carbonate. Tube worms appear, their tubes pale and clustered, their plumes extended into the current. The camera moves slowly over them. The worms do not know they are being filmed. They are engaged in a more ancient activity, exchanging chemicals with their symbionts, drawing oxygen from water and sulfide from below, keeping alive the partnership that makes their bodies possible. Nearby, mussels form a dark mound. Their shells are edged with sediment. Small animals move among them. The bed looks still until the camera stays long enough to see motion. A shell opening, a shrimp flicking away, a crab picking carefully, a worm bending. Deep sea time reveals itself only to attention that waits. The vehicle stops and hovers. For a moment, no one speaks. There are times when narration should become thin. The sea floor does not need constant explanation. It needs space. Bubbles rise. White mats shift. The current carries fine particles sideways. The lights illuminate a few meters of a world that continues far beyond them. This is the center of the film, whether it appears at the beginning or the end. A small leak and an entire hidden logic around it. The leak says the sediment is not inert. The mats say microbes are working. The mussels say chemistry has become food. The carbonate says the process has lasted. The bubbles say some methane escapes despite the filters. The water column says the story is not over. The atmosphere far above may or may not ever meet this methane. That depends on depth, dissolution, oxidation, currents, and time. But even if no molecule from this seep reaches the air as methane, the seep still matters. It matters because the ocean's carbon cycle is not only about what reaches us. It matters because life has made a world here. It matters because deep water chemistry is changed. It matters because the sea floor's hidden processes are part of the planet's actual functioning, not a footnote to human concerns.
This is a difficult lesson. Something can matter without being centered on us. A seep does not have to threaten a city to deserve understanding. It does not have to become fuel to have value. It does not have to resemble a coral reef to be beautiful. It does not have to be visible from space to be part of Earth's story. At the same time, it is not separate from us. The methane cycle connects to climate. Offshore industries connect to seep habitats. Ocean warming connects to hydrate stability. Our energy history connects to the same hydrocarbons that feed some seep ecosystems. The deep sea is not about us, but we are entangled with it. The ROV eventually turns away from the seep. The dive has a schedule. The ship has fuel limits. Weather is coming. Another target waits. The vehicle rises a few meters, then 10. The scene begins to flatten. The worms become pale strokes again. The mussels become dark roughness. The bubbles become bright flecks in the lights. Then the bottom disappears. During the ascent, the water outside the camera looks empty again. But now that emptiness has changed. It is no longer blank. It contains dissolved gases, microbes, particles, currents, chemical memory. Somewhere below, the seep continues. Somewhere above, the surface waits, unaware of the small transformations happening beneath it. The ship recovers the vehicle hours later. People on deck guide it in with lines. Water drains from the frame. Samples are carried away. Screens are shut down. The ocean around the ship looks ordinary, a dark, moving surface under weather and stars.
This is another contradiction of methane seeps. The surface gives almost nothing away. A person standing on deck cannot see the leak below. A satellite cannot see most deep bubbles. A coastline cannot reveal the chemical habitats offshore. The ocean keeps a calm face over intricate interiors. Maybe that is why the subject feels like a mystery. Not because the science is absent, but because the evidence is hidden behind a surface that looks complete.
The sea has always been good at hiding its depths. For centuries, people imagined monsters, gods, lost cities, and bottomless abysses. Modern science replaced many myths with measurements, but the replacement did not make the ocean smaller. It made the unknown more specific. Instead of vague darkness, we now know there are methane seeps, hydrate fields, microbial filters, brine pools, trench communities, and thousands of unmapped places where chemistry may be feeding life. The unknown has become more detailed. That is progress of a profound kind. It means the mystery is no longer empty. It has texture. It has names, instruments, organisms, depths, rates, and unresolved questions. The old blank has become a landscape still waiting to be fully seen. At a methane seep, this landscape is not only beneath the ocean, it is beneath the way we usually think. It asks us to look past surfaces, past categories, past the separation between living and non-living systems. It asks us to imagine the planet as a series of exchanges, many of them slow, many of them microbial, many of them hidden until a bubble catches the light. When the final image comes, let it be simple. Not a map covered in points, not a graph, not a warning title. A small patch of seafloor, cold water, mud, shells, a pale bacterial veil, a few bubbles leaving the sediment one after another, rising into darkness, shrinking, dissolving, becoming something else. That is the story in miniature. The earth releases, the ocean receives. Life intervenes. Nothing remains only what it was. There are stretches of the ocean where a seep may exist for a long time as rumor before it becomes confirmation. A faint anomaly in old sonar, a report of unusual backscatter, a chemical spike in a water sample, a pockmark on a bathymetric grid, a cluster of hard returns where the seafloor should be soft. Each clue alone is uncertain, but together they begin to pull attention toward one place. This is how much of the deep ocean is approached, not as a landscape already known, but as a set of hints. The ship does not sail to certainty. It sails to probability. It lowers instruments into a question, and sometimes the question answers with bubbles. Other times, nothing appears. The suspected seep is quiet, or the bubbles have stopped, or the acoustic signal was something else, or the leak is diffuse and invisible. These failed dives are part of the story too. They remind us that the seafloor is not obliged to match our interpretations. A negative result can still teach. It can refine methods, correct assumptions, redirect attention, and reveal how much variability lies between surveys. A world of seeps is also a world of false starts. That gives the confirmed places more weight. When bubbles are actually seen, when mussels are actually found, when methane concentrations rise in samples, when carbonate carries the isotopic mark of methane-derived carbon, the place becomes more than a possibility. It becomes part of the known seafloor. Known, but not exhausted. A named seep is not a solved seep. It can be visited many times and still hold surprises. One year, a bacterial mat may spread. Another year, it may shrink.
A mussel bed may show recruitment of juveniles. A carbonate outcrop may reveal corals not previously noted. A plume may strengthen after a change in sediment pressure. A microbial community may shift with flow. The name helps people return, but returning shows that the place is alive with change. This is especially true where seeps lie near human activity. In the Gulf of Mexico, natural seepage and industrial hydrocarbon extraction exist in the same broad region. Distinguishing natural emissions from human-caused releases can be critical after accidents. Understanding background seepage helps interpret anomalies. Baseline seep maps are not only ecological tools, they are forensic tools for a sea increasingly shaped by infrastructure. The deep ocean is no longer beyond industry. Pipelines cross it, wells enter it, cables span it. Sound from ships fills it. Trawls scrape parts of it. Proposals from mining reach toward it. Research vessels study it. The idea of an untouched deep sea is fading. What remains is the harder idea of a deep sea that is still wild in many ways, still poorly known, but no longer isolated from human systems.
Methane seeps make this entanglement visible because they sit at the intersection of hydrocarbons and habitats. The same substances that power modern economies can feed deep-sea life when released naturally in the right conditions. This does not make fossil fuel use benign. It makes the carbon story more layered. Methane is not one thing in one place. Context changes consequence. At a seep, hydrocarbons are not extracted. They are metabolized, filtered, and woven into ecosystems. In industry, hydrocarbons are removed, burned, and added to the atmosphere in enormous quantities. Confusing these contexts would be a mistake, but recognizing their connection can deepen the sense of irony. The gas rising gently from a seep is kin to the gas moving through pipelines, but its path, scale, and fate are different. The planet's carbon has always moved. Human beings have changed the rate, route, and volume of some of that movement. Natural methane seeps remind us that not all emissions begin with people, while climate change reminds us that natural systems can be pushed by human forcing. The distinction between natural and human-caused remains important, but it does not create a wall. A warming ocean can influence natural reservoirs. Human disturbance can damage natural habitats. Natural microbial filters can reduce methane release, but their capacity is not a license for complacency. The seep teaches connection without erasing responsibility. This is a subtle lesson, and subtle lessons are easily lost. They do not make clean slogans. They require duration. They require the kind of long-form attention that can hold a bubble and a climate model in the same frame without pretending they are the same thing. Maybe that is why the ocean floor's methane leaks are worth following for so long. They reward patience. The longer one stays, the more contradictions settle into coherence. The leak is danger and nourishment. The deep is remote and connected. The gas is natural and climate relevant. The habitat is harsh and abundant. The bottom is an ending and a beginning. By the time the camera returns to the first bubble, it is no longer just a bubble. It is a sign of buried microbes, a path through sediment, a possible hydrate boundary, a meal for bacteria, a question for climate, a clue for geologists, a sound for hydrophones, a shimmer for cameras, a moment in a carbon cycle far older than human language, and still, it is only a bubble. This double vision may be the most honest way to see the deep sea.
Let the thing remain itself while also allowing it to connect outward. Do not crush it under symbolism. Do not reduce it to a fact. Keep both scales alive. The cold seep remains mud and chemistry, animal and planet, dot on a map and world unto itself. In the darkness, no conclusion is required. The seep does not need the story to finish. It will continue until its pathway closes, until pressure shifts, until hydrate forms or breaks, until flow migrates, until sediment buries one opening and another appears. The community will follow if it can. The microbes will respond. The carbonate will stay behind. The ocean will carry away what dissolves. A person watching from far above can only imagine the continuity, but imagination, when grounded in evidence, is a form of attention, and attention is where care begins. Far from the cameras, in seep sediments that have never been sampled, the same quiet negotiations may be taking place. Methane moving upward, sulfate moving downward. Microbes meeting in the dark between them. A chemical boundary only a few centimeters thick may decide whether gas escapes or becomes food, whether carbon enters water or rock, whether a pale mat appears on the surface or the mud remains visually plain. This hidden thinness is astonishing. So much can depend on a layer no hand could separate cleanly without changing it. The ocean floor looks broad and solid, but the crucial action may occur in films, pores, gradients, and surfaces. The deep sea is vast, yet many of its decisions are microscopic. Above those microscopic decisions, animals wait for conditions to become livable. Not comfortable, livable. A larva settles where the chemical signal is right. A bacterial partner colonizes tissue. A worm tube lengthens. A shell thickens. A mat spreads over mud. What appears to humans as a strange community is, to the organisms, a set of precise solutions to physical problems. How to eat without sunlight. How to breathe where sulfide is toxic. How to stay close to methane without being overwhelmed by the chemistry around it. How to build a body where food is made inside you. How to endure in a place that changes slowly until it changes completely. The seep answers through form. A tube worm's root system, a muscle's gill, a clam's foot, a bacterial filament, a carbonate ledge. Evolution has shaped each one as a response to the leak. Perhaps that is why these habitats feel so intimate despite their remoteness. They are not random assemblages. They are communities fitted to a particular opening in the planet. Remove the opening, and the pattern changes. Move the flow, and life rearranges. The seep is not just where they live. It is the condition of their form. At the surface, waves may be rising. Weather may be crossing the ship. Satellites may be tracking cloud bands. People on land may be sleeping in cities powered partly by methane from ancient reservoirs. None of that is visible below. The seep continues in its own register, linked to the surface by chemistry, but not by awareness. The ocean is full of such asymmetries, things connected without knowing each other. A storm alters pressure over a shelf. A current shifts bottom water temperature. A plankton bloom months earlier adds organic matter to sediments. A warming climate changes the stability of frozen carbon. A bubble dissolves before reaching sunlight. The connections are real, but they do not resemble human relationships. They are physical, delayed, and often one-sided. To understand the sea, the mind must accept connection without closeness. Methane seeps are distant, but not disconnected. They exist at the bottom of the ocean and inside the global carbon story. They are remote habitats and active chemical processes. They are places a person may never see, and places whose behavior may matter to the atmosphere above every person. This combination is difficult to feel But the image of the bubble helps. It gives distance a body. A bubble is small enough to follow. It is also connected to everything. So the story narrows again. Not because the larger map has vanished, but because the map is held inside the smallness now. The bubble lifts from mud. The mat glows pale beside it. The muscles remain closed until one opens slightly in the current. The camera light trembles with the vehicle. Beyond the light, the ocean is black. Below the mud, methane keeps finding its way upward through the planet's imperfect seal. No one standing on a beach would know it is happening. The surface keeps its ordinary language of waves, gulls, wind, and light. Beneath that, beyond the shelf and down the slope, another language continues without sound a human ear can catch. Methane, sulfate, carbonate, sulfide, shell, microbe, current, pressure. The words are ours. The process is not. It has been moving through the seafloor since long before anyone named the ocean, and it will keep moving after the latest map is replaced by a better one. Each new survey will add points. Each dive will add detail. Each sample will sharpen the chemistry. But the dark between the points will remain, holding more leaks, more communities, more patient exchanges. The ocean floor is still not fully known. It is still opening in small places, still feeding life from below, still taking what rises and changing it before it can become something else. And in that unfinished exchange, the deep sea keeps its calmest mystery. Not a silence, but a slow conversation between buried carbon, cold water, stone, and living cells. Enough.